curates and disseminates published 3D structural data in the form of the Cambridge Structure Database. But we've also worked and partnered with industry and academia to accelerate both the discovery and the development of medicines and materials, as well as doing some extensive outreach and education to the global scientific community. So I thought I would introduce the concept of data-inspired medicines design by reminding everyone that behind every medicine that has really helped and advanced humankind, there is a crystal. And for discovery scientists, it's fairly well understood that X-ray crystallography is vital to structure elucidation, establishing what the molecular structure is, what the stereochemistry is, the molecular conformation, and the like, not just in the drug crystals themselves, but in receptor sites. But what a lot of people don't maybe understand is that crystal chemistry perhaps plays an even more important role in drug development, because the molecules are genuinely, for at least solid oral dosage forms, delivered in crystalline forms. And so there is an enormous amount of science that goes on trying to determine how molecules crystallize in different forms. And because they are because it is possible for molecules to crystallize in different forms, a very important part of drug development is to establish how a molecule will crystallize. This is generally the first step in drug development, determining how a molecule can be isolated in the crystalline state, hopefully purified as part of the crystallization process, and used to ultimately deliver the drug to the patient. And of course, any product or process that we intend to design can only be designed for and around the crystal forms that we know about. And so this means that a lot of attention has been paid to determining how molecules will crystallize, hoping that even one of those crystalline forms will be suitable for a commercial drug product. And so the industry has long been aware of the importance of solid state chemistry in development, but the world pretty much changed in the late 90s with ritonavir in terms of the level of interest that the pharmaceutical industry took in crystal chemistry. And this was an instance where a drug product by the name of Norvir, which was used to treat HIV and AIDS patients, was marketed in polymorph form 1. And the compound was in the market, the product was successful until some 240 batches into the marketing, or the scaled up marketing endeavors, that a new polymorph appeared, and this polymorph caused dissolution failures that had not been seen. That is because this polymorph form 2 was more stable and less soluble than the marketed crystal form 1. The impact of this late appearing polymorph was that the Abbott company at the time had to withdraw this medicine from the market while they reformulated. And it caused not just hundreds of millions of dollars of lost revenue to the company, but it also forced the removal of a medicine from the patients who really needed that. So this was really a wake-up call to the pharmaceutical industry that we could not have another market recall due to not having control over the polymorph landscape of our drug molecule. The problem is that we can screen for solid forms, but there really is no way to predict how molecules will choose to crystallize. And in this example, I'm showing two lily molecules that I worked on in my previous life that highlight some of the challenges. Here we have two molecules in a series that were clearly developed for the same application. and they look very similar, yet their crystallization behaviors were entirely different. And we would never have known a priori that the biggest challenge in developing the compound on the left would be figuring out which of two relatively highly crystalline forms was the most stable form. Contrast that to the molecule on the right, where the challenge was finding any crystalline form at all. So clearly, you cannot look at a molecule and determine how it will crystallize, if it will crystallize, and how many forms it will crystallize in. Now, this is something that actually I looked at in collaboration with Aurora Cruz-Cabeza and Joel Bernstein a few years ago. We were asking ourselves, what is the prevalence of polymorphism in well-studied molecules? And so I compiled 15 years of statistics on the right from screens that I had conducted or overseen at Lilly, with some of the better compilations of polymorph appearance that were in the literature. And the take-home message here is that polymorph appearance is very prevalent. And in fact, 80% of all of the compounds that we investigated had more than one form, polymorphs being more prevalent among parent compounds, hydrates becoming more prevalent among the salt options. The work that we had done that culminated in the facts and fictions work with Aurora and Joel really started to look at molecular underpinnings for polymorphism. And to make this happen, Aurora, who is an expert in curating CSD, Cambridge Structured Database structures, was able to create data sets for compounds that did not show polymorphism and also data set that did show polymorphism. However, we were really concerned about the bias in that sort of data, because the data that is collected in the CSD comes from all sorts of different studies, not necessarily intending to seek out polymorphism. What Aurora came up with, which was really quite clever to make sure that we removed as much as we could the bias in the data set, was to consider monomorphic systems for which there had been two structure determinations in the CSD. And that would essentially guarantee that those molecules would have at least been crystallized twice, which is really not a lot to ask, but asking that much removed the bias from our data sets so that we could now look at polymorph occurrence for molecules that were considered polymorphic or were not considered polymorphic. And so we combined data from the CSD as well as Lilly when I was there, and Aurora was postdocing at Hoffman-LaRoche at the time, and we were able to look at the prevalence of polymorphism in light of a number of molecular attributes shown here to the bottom. The end game and the take-home message here was that there were no molecular properties, perhaps with the exception of chirality, that would bias a molecule toward forming polymorphs or not. And this was very important for a lot of the patent litigations around crystal forms to support the claims of non-obviousness of crystal forms. There is just simply no way to look at a molecule and its structure to determine whether or not polymorphism would be likely to occur. So a lot of screening went on. A lot of automated platforms were developed by the pharmaceutical industry following the ritonavir crisis. And the idea was that maybe the automated platforms could maybe help us de-risk solid form selection. So I want to break the rest of this presentation into three general areas for de-risking solid form selection, starting with experimental solid form screening. So experimental solid form screening basically amounts to exposing molecules to a variety of different crystallization conditions, hoping to trigger different nucleation events. And so a lot of automated platforms have been developed over many years that can accommodate hundreds, if not thousands, of crystallization experiments in parallel. These high throughput high throughput techniques were believed to be the panacea for finding polymorphism. However, in my own experience, high throughput screening would definitely fail if you didn't consider the solubility properties in the diverse solvent systems you use in screening, or if you would not take into account the highly different crystallization kinetics. So there are definitely failure modes to the high throughput screening. So we can ask ourselves, how did we do? And what I would say is that 10 years after the ritonavir crisis came the rotigotine crisis. And this happened when the rotigotine patches, part of the NoiPro product developed and commercialized by UCB Pharma, experienced a similar fate as ritonavir. And you can see the transdermal patches to the right show these snowflake-like crystals growing. And those crystals were of a new polymorphic form, form 2. What this tells us some 10 years after the ritonavir crisis is that polymorphism is not a solved problem, and that the experiments that were created en masse through the automation were simply not enough. Now, it's not a knock on the automation. The automation did exactly the experiments that the scientists told them to do. It's really more a matter of where our understanding of crystal nucleation and growth is. I would venture to say that it was because the scientists didn't know what experiments to tell the robots to execute, and therein lies the problem. So we need something more than the experiments. And to highlight one of the real challenges to appreciate why you can't just design a one-size-fits-all screen to find all your forms, I'd like to just kind of share a bit on the history of aspirin. Aspirin has been manufactured in metric tons for decades and for a long time was believed to only form one polymorph. But then there was a report of a second polymorph, form 2, which turned out to be rather controversial. And finally, Andrew Bond and his team did some really elegant work to show that there was a second polymorph, but it was actually grown as intergrowths in the form 1. It turns out that there was really no way to produce form 2 in a stable form by itself, free of form 1, until Andrew Bond and his team crystallized aspirin in the presence of aspirin anhydride. And this anhydride allowed form 2 to be produced free of form 1 and stable for some period of time. Later on, not that long ago, Bart Carr and his team out of NYU produced crystal form 4 of aspirin. In this case, he crystallized the new polymorph from a melt from a gummy material by the name of Canada balsam. And I use this example as a clear case where there is no way to build in the effects of different impurities on crystallization in any sort of crystallization design. So we really have to move beyond trial and error experiments if we are to assure ourselves that we have found the most stable form and are at less risk of something else compromising our product. This has really forced us to turn toward computational techniques for one, starting with crystal structure prediction. Crystal structure prediction itself is a full seminar, but I just want to say at a high level what CSP is, and that is taking a two-dimensional molecular diagram and calculating the different ways that that molecule can pack in a crystal structure. There will be hundreds of thousands, even millions of crystal packing possibilities that are calculated in these computationally intensive workflows. And so with that many structures that are being predicted, the energy model that you would use tends to be the most affordable energy model that you can apply. The idea then is in a second energy ranking or re-ranking step that you would take the lowest energy structures among the batch and apply a better energy model to produce a 0K lattice energy with a great focus on the lowest energy structures. So that is CSP, but why are we interested in CSP? And I would venture to say that we've paid attention to crystal structure prediction probably for as long as we knew it existed, because we spent a lot of years with trial and error experimentation, not knowing when to stop an experimental form screen or to continue looking for something else. So we posed the question in our product design challenge of what would happen if we actually knew the hypothetical structures that should be experimentally accessible, and what if we knew their solid state properties as well as the conditions to produce seed crystals. If we knew all of that from a computer-aided design standpoint, then we would only ever target those crystal forms with the properties amenable to delivering a drug. And if we could do that, it would be it would offer enormous savings to the solid state form footprint of commercial drug development. So we are very interested in crystal structure prediction and have been tracking the progress of the CSP algorithm development over many years. It wasn't until the 5th blind test, which was published in around 2011, that I truly got excited about the prospects of crystal structure prediction. And here I would draw your attention to molecule 20 in the bottom center. This, as each of the blind tests had progressed over the years, the substrates would get larger and more complex. And it wasn't until molecule 20 that the molecules started to look like the smaller sized molecules I had seen in drug development. So I was really excited that the methods might actually be ready to be able to apply to the drug development scenario. What you can see from this slide is at the time, only 10 groups in the world attempted the crystal structure prediction of molecule 20, and only 2 of them got it right. One of those 2 groups was the group of Sally Price out of University College London. And it was at this point that I phoned Sally up and asked her if she would be willing to collaborate on a crystal structure prediction project looking at well-characterized Lilly molecules so that we could see how CSP played out in a real drug development setting. And so that started what was better than a 10-year collaboration with Sally Price. And the very first system that we worked on was a former Lilly blockbuster drug by the name of olanzapine. And what you have here in this center plot is referred to as a crystal energy landscape. Each one of the data points on this landscape corresponds to a thermodynamically and mechanically stable crystal packing. And these packings are sorted. They're certainly ranked by energy, so we focus on the low energy structures or data points. And they're separated, in this case, by packing index or density. The structures could be separated by really any metric that you wanted, but it's quite common to separate the structures out based on some sort of close packing metric. What you can see from this particular slide is that we were able to identify, out of the 3 known polymorphs of olanzapine at that time, crystal structure models for those polymorphs, form 1 being the most stable structure, metastable polymorphs 2 and 3 always crystallizing together, crystallizing concomitantly. We were lucky enough to eventually pluck a single crystal of form 2 out of a mixed form 2 and 3 batch and get its crystal structure. And once we knew what that structure was, we could identify the unaccounted for peaks and come up with a structure model from CSP that allowed us to propose what the crystal structure of form 3 was. What's really interesting about olanzapine is that it's a highly crystallizable molecule. There are 56 at least crystal structures of the parent compound. And every single one of those crystal structures is based on a dispersion-bound olanzapine dimer. And those are exemplified by these red diamonds. But what you can see in Sally Price's crystal structure prediction is that there are many structures that are not based on that dimer. And that prompted a lot of work by many groups around the world to try to find some of these non-dimer based structures, some of which were predicted to potentially be more stable than the known form. It wasn't until about 2019 that a group in the UK accidentally or serendipitously did discover a non-dimer based crystal form 4. And this was based out of work intended to look at amorphous solid dispersions of olanzapine. So the drug was actually embedded in a polymer, which was apparently enough to break up the dispersion bound dimer, which allowed form 4 to uh, to finally nucleate and eventually grow. And so this was fantastic, and we were super delighted when Erin Johnson and her team at Dalhousie State in Canada picked up the olanzapine system and extended the energy modeling from the models that we had published on to using much more sophisticated molecules. And we were absolutely delighted that the more expensive molecules showed that in terms of lattice, 0K lattice energy, the 4 observed crystal forms corresponded to the 4 most stable structures. Olanzapine has spurred a lot of interest even more recently, and a group out of China extended the energy analysis to also look at free energies under the harmonic approximation. And so what this really has helped us to appreciate is what is the relative stability of these forms, not just at 0 degrees Kelvin, but at crystallization process and storage relevant temperatures. And these these free energy curves very nicely agreed with the stability order that we had come to know experimentally. Now, Sally and I continued in our collaboration to look at a variety of different systems, and we learned an awful lot in the process about how to approach interpreting these very complicated crystal structure landscapes, where clearly many more structures are predicted than ever we would observe. Now, we use these landscapes similar to an olanzapine to help us solve crystal structures by providing suitable structure models that would allow us to solve structures from, for example, powder diffraction patterns. We learned how to deal with disorder and how to interpret systems that showed signs of disorder. We proved to ourselves that we were fully able to generate high energy crystal forms experimentally, and once again reminded ourselves how non-obvious the crystallization behaviors are, because the molecule on the right and left basically possess the same hydrogen bonding functionality, yet all thermodynamically, or most all of the thermodynamically stable structures on the molecule on the left are based on intramolecular hydrogen bonding, whereas many of, if not most, of the stable structures on the right are based on intermolecular hydrogen bonding. So this really goes to show that molecules that are clearly targeted for the same receptor site in the body may have entirely different crystallization behaviors. One of the last systems that we've worked on, Sally and her team and I, was a compound by the name of galernicetib. This is a decamorphic system. Actually, this molecule, as of 2019, had set a world record in terms of the number of fully structurally characterized highly polymorphic systems, a record that has since been eclipsed by the famous Roy molecule. But we used galernicetib as a test case to study how CSP could be used in a development setting, and also to bring the capabilities in-house to Eli Lilly and Company. Now, why were we so interested in CSP of galernicetib? And the answer is quite simply that we had some real hurdles in the experimental solid form screening of the galernicetib molecule. In this system, anytime galernicetib came in contact with water, we would form a highly stable crystalline monohydrate structure. So unless we took measures to get water out of the experiment, this is really the only structure we would tend to see. That certainly provided some real limitations in terms of the kinds of crystallization experiments we might do in screening the galernicetib molecule. Similarly, at high temperatures, galernicetib proved to be chemically labile. It fell apart. And so that removed another major avenue for achieving crystal nucleation conditions that promote diversity. And then when we moved to solvent-based screening, we soon found out that galernicitib formed solvates. And it formed a lot of solvates, wherein different solvents would be incorporated in different crystal structures of the molecule. And so when you're looking for non-solvated crystal forms or hydrates to develop for a commercial drug product, oftentimes with solvates, you need to work around those crystal forms to find the structures that you would care about from a commercialization standpoint. The problem is when you have this many structures, you have to be rather creative to find conditions where the parent compound would not form a solvate. And so here's where crystal structure prediction can be so helpful, because it can show us theoretically what non-solvated crystal structures might be out there that might be stable, that might motivate us to continue our search for commercializable crystal forms. So Sally Price and her team generated a crystal energy landscape of the galernicitib molecule. And once again, you see many, many different crystal structures. And even though I've given you the answer in the sense that there are 10 polymorphic forms that we have observed, at least to date, you can see there are many more structures. We were able to see the stable crystal forms that we could crystallize directly from solution on the landscape, as well as some of the neat desolvate polymorphs that we had known to date. But what we were really surprised about was this structure at the lower right, which was far more dense than the experimentally observed forms, but more importantly, possibly far more stable than any of the experimentally observed forms. And so when we saw that purported crystal structure, that putative polymorph, we went back into the laboratory to extend our experimental form screening. And that's when we discovered forms 7, 8, and 9. But forms 7, 8, and 9 were not this global energy minimum structure to the lower right. So we extended further our experimentation to include high pressure crystallization, extending our collaboration with Sally Price to include Colin Pullum out of the University of Edinburgh. And so Colin and his team had used some conventional techniques to crystallize galernicitib under pressure, starting with direct compression of the known polymorphs as well as solution crystallization. And in no instances did we see a crystal form by those methods that we had not already seen. One of the observations that was interesting to us, however, was that under pressure, galernicitib seemed to be less thermally labile than it had in our conventional laboratory experiments. And that is consistent with some of the early reports that thermal decomposition could be suppressed at high pressure under nitrogen. So this motivated us to pursue melt crystallization. Remember, we couldn't really sample temperature in our experimental solid form screen at ambient pressure. To make these experiments work, we selected as a starting material the lowest melting form that in our hands was shelf stable, and that was form 5. This would allow us to achieve the melt under the lowest possible temperature. And so when we did that, Colin and his team grew this very large single crystal that I'm showing the picture of in the upper right in the diamond anvil cell. And it turns out that this structure did survive when we decompressed it, and we were able to get a crystal structure. And lo and behold, we now had the 10th polymorph and an X-ray crystal structure of it. What's interesting is that form 10 was not the global energy minimum. In fact, form 10 is actually in the middle of the landscape. And so this is just a nice, useful reminder to us that we can't yet go into the laboratory and select for any given one of these putative structures. And that's really where the state of the art is taking us. Now, I do want to say that we were looking at bringing crystal structure prediction methodology in-house, and galernicitib was one of our first cases where we were trying to get Marcus Neumann's grace code up and running. And so Jonas Nyman, who was postdocing for me at the time, calculated the crystal energy landscape of galonicitib, extending the search to Z prime of 2 structures. And one of the things that was really of interest to us was that exactly the same global minimum structure that Sally Price and her team had predicted was seen on this landscape. However, with a different energy model, you can see that it's not so much more stable, at least in terms of 0K lattice energies, as it was with the previous model. And so I'm going to come back to the galonicitib a little bit later, but what I really want to say is that the experience that we've gained in the better part of the decade has certainly taken us to bring crystal structure prediction in-house, and this is very consistent with what we're seeing across the pharmaceutical industry, where CSP is now fairly widely adopted. And the CCDC is actually responding to the call by making a product line called CSD Theory come to life. And this is basically software for the storage, the visualization, as well as the management of CSP knowledge. But where are we now today, aside from the data side in structure prediction? Because we have all of these landscapes and a clear issue of overprediction. Well, I'm outlining 3 different scenarios of where we kind of are today. And the first one is shown on the upper left with crizotinib, which is a Pfizer molecule. In this instance, the experimental structure is shown in the green square, and it is much more stable than any other predicted structure, which tells you that that molecule is really quite happily interacting in one specific 3-dimensional crystal structure environment with itself. And this is the ideal situation in terms of assessing polymorph risk and providing the assurance that maybe there isn't another energetically competitive structure out there. The more common situation, however, is shown to the right with dalcetrapib, where there are a variety of energetically competitive structures. And this was some work that Marcus Neumann and his group had published a few years ago that motivated high pressure crystallization and the discovery of an energetically competitive form, which happened to be disordered. So the 3 diamonds actually correspond to the 3 ordered components. But like the previous example, we can't just use pressure or any of these other techniques to dial into any given structure. Probably the most challenging situation, and I do think this is rare, but it is interesting, and that's with this melatonin agonist compound on the lower left. This was a molecule that was under development many years ago, and early in development, there was not very much material available. And so a lot of the solid form work was conducted using the opposite enantiomer. And so the polymorph screening on the opposite enantiomer did yield the most stable form. In fact, years later, there was a CSP study which showed that that was, in fact, the most stable form. However, when my colleague moved to the correct enantiomer, the one that was intended for the drug product, he and his team could never produce the enantiomer in that most stable structure, no matter what they did. So this was not a case where the most stable structure couldn't crystallize. In fact, actually it had just of the wrong enantiomer. So there is, in theory, a way to get that crystal structure. We just didn't know experimentally how to do that. In this case, when you know that one of your putative structures actually can exist and that it is more stable, you can start backing out what happens with respect to, for example, solubility by looking at the energy differences between your between your calculated structures, and in this sort of situation you would formulate hopefully to the least soluble components so that you don't run into a ritonavir or a rotigotine case should that more stable form at some point decide that it would appear. So I want to turn to the third part of de-risking solid form selection. We've gone through the experimental screening challenges and the promise of crystal structure prediction, but we still have this issue of over-prediction and under-finding polymorphs. And so how do we begin to interpret the crystal energy landscapes to truly assess what the risk of one of these putative structures, if they are in fact more stable than the known forms, what that risk might be. And for this, we turn to solid form informatics, which are basically data-driven approaches. And when I say data-driven, I'm really referring to crystal structure data-driven. And this brings me to really more about my interest in joining the Cambridge Crystallographic Data Center. So we were really interested in data-driven approaches using a wealth of crystallographic data based on some of the inspiration of one of our founders, Olga Kennard, who passionately believed that the collective use of data would lead to the discovery of new molecules above any of the individual experiments. And so the data-based approaches that I'm referring to are built on the Cambridge Structural Database, which now stands at more than 1.1 million structures. This database is the largest database of published structures in the world, containing, as I'll show you on the next slide, a variety of organic and metal organic structures. This database is enriched by experts and highly curated, and it's really been built on a community effort of almost 60 years to get to this number of structures. It's really exciting to see the growth of crystallography mirrored in the growth of the Cambridge Structure Database. When we look into the database, we see molecules of significant commercial interest. We see a tremendous amount of diversity in the structures, and we see an awful lot of extra data that can be used in different ways to start relating structures and properties. And the CCDC has done more along these lines to create subsets of the CSD to help target certain applications in a variety of different sectors. So what is it about the data-based approaches that will help us and aid us in solid state form design? Well, let's start with understanding molecular shapes. What we find is that for any given molecule, we can interrogate using the Mogul engine the bond lengths, angles, and torsion angles, and the statistical distribution of structures that have those particular groups. And what you can see is that with enough data, that the distribution of, in this case, torsion angles very much mirrors what you might expect to see based on computationally derived torsional energy profiles. The beauty of the data approaches is that we can generate histograms based on CST data within seconds that might take days to weeks for molecules using, for example, QM methods. So there's a lot of interest in the medicinal chemistry community, in drug discovery in particular, to be using these data-based approaches to rapidly survey many different compounds that might be entertained in establishing, for example, structure activity relationships. In terms of intermolecular geometries, we have within the superstar framework the ability to look at intermolecular interactions about specific functional groups. From various crystal structures, we can look, for example, at different probes to look at where the close contacts are and what those geometries, in fact, are among all of the relevant crystal structures in the CSD. If we put this information together, if, for example, we start looking at a combination of the different flexible torsions in our molecules, we can start to back out what the likely crystal conformers will be. If we choose to look at how the functional groups in our molecules might hydrogen bond with their neighbors, we can start now to pull out full interaction maps showing the distribution of interactions around the molecule of interest. And so this allows us to take millions of data points around intramolecular geometries, intermolecular interactions, and crystal packing, and to ask ourselves for our structure, how can we learn about how our molecule will crystallize based on the more than 1.1 million structures in the CSD that already have? We can begin to ask ourselves how the shape of the molecule compares relative to the statistical distribution. Is our crystal conformation strained? We can start to ask ourselves about how the molecules self-assemble by, for example, hydrogen bonding. What are the pairings? What is the coordination? How is the geometry influenced by crystal packing? And we can even look at what's not in our crystal structure, looking at the space, the void space in the crystal, which oftentimes can translate into chemical liabilities or even unwanted moisture absorption properties. So these sorts of checks that we have built tools for are now linked into workflows that allow us to establish the health, if you will, of any given crystal structure. And these health check workflows are now being increasingly widely applied across the pharmaceutical industry. Two very quick examples. This was a case of a compound by the name of Maraviroc, which was a Pfizer compound. It was first isolated in form A, and the health check that was performed on this quickly revealed that the hydrogen bonding in form A appeared to be suboptimal. This triggered more experimental screening, which led to a more stable crystal form B. And so this is really your idealized scenario where the informatics approach provided the impetus to do more experimental screening. Potentially a more realistic case, however, shows some of the compromises that molecules are willing to make when they crystallize. In this instance, also a Pfizer compound, you would see different hydrogen bonding patterns between the two known crystal forms, forms A and B. And the late appearing form did seem to have better hydrogen bond geometry. However, you can see in terms of the molecular shape on the right that the crystal conformer was quite unusual. This can raise a lot of concerns about how easy it might be to attain a crystal conformation in solution or in the gas phase. And could this in part explain the late appearance of form B? I want to take us back as we close this presentation out to gallernicitib, because we had the 10 known polymorphs, but clearly there was this global energy minimum structure, which despite extensive screening from many groups around the world, could not produce that structure. The very first thing that one does when it appears that there is a more stable unobserved structure is to check the energy models. And we, in fact, had done that prior to our 2019 publication. And what we were able to show was that maybe the global energy minimum was not quite so much more stable than the observed forms, but it was certainly energetically competitive. And we had published this work, we had not done free energy calculations at this point, but that remains to be seen. And so what approach we took was maybe a little bit different, and that was looking at conformational strain energy as a surrogate to crystallizability. The idea being that the more strained the molecular conformation, the less likely that crystal conformer is to predominate in crystallizing solutions. What you can see in the table to the right is that the global energy minimum structure was the most strained crystal conformer of any of the experimentally observed plus global minimum structures that we had. However, the strain energy was not so much higher than at least one of the conformers in form 7 to rule out that you could not see it. We knew that Form 7 was derived by desolvation under mild conditions. And so could there be a solvated precursor of that could potentially take us to the global minimum structure? To address that, we started to look at the structural data that we had accumulated for the galenicitib molecule. We knew that galenicitib was a prolific solvate former, and we had quite a few crystal structures of the solvates themselves. And we could see that some of the solvates bore structural relationships to the neat crystal forms that could be obtained under mild conditions. But what you see in the very lower left is that the global minimum structure has no resemblance to any known structures. So it really was not clear to us that there was any sort of solvate precursor that might seamlessly desolvate to that crystal structure. We then applied the crystal structure informatics health checks, starting with hydrogen bond propensity. And this is a CCDC tool that has been used to look at donor-acceptor pairings of different hydrogen bonding sites. With the galenicitib molecule, we have 2 amide NH donors competing really for 4 different acceptors. And among these acceptors, the quinoline, which is nitrogen 1, is probably the best acceptor. But the second best acceptor is arguably pyridine nitrogen 4. And the second best acceptor is almost never used, except in one of those conformers in Form 7. Yet if you consider the global minimum structure at the bottom, the hydrogen bond to the pyridine nitrogen 4 appears to be the strongest and the best pairing in that structure, which was really highly unusual. What was also unusual came into focus with analysis of the full interaction map. Here we are looking at the interactions for the galenicitib molecule in the global energy minimum structure. And this allows us to look at how well satisfied the hydrogen bonding interactions are. And what you can see around nitrogen 4 in the middle of the molecule is there really is no hot spot where the donor nitrogen is. And that's because this pyridine ring is inward facing. It is not sterically accessible to hydrogen bond donating groups. Hence, in most of the crystal conformers, that site is never used. So here we have, in our global energy minimum structure, a strange crystal conformer with far from ideal hydrogen bonding interactions and no clear parent solvate structure with which to cleanly desolvate to. And it was this body of information that convinced us that even though this structure is energetically competitive, it might be unlikely to see this structure on the timescales of drug development. So I would like to bring this lecture to a close to hopefully highlight the different ways between experimentation, computational chemistry, and informatics to assess the risk of solid forms, and to also paint maybe a bigger picture around what it takes to inspire data or to use data to inspire medicine's design. We've seen examples in this presentation of using solid form informatics, and we have a lot of activity going on developing particle informatics tools as well outside the scope of this presentation. And we also are increasingly interested in machine learning applications to derive material properties. So we believe material informatics has an important role to play alongside experimentation and alongside computational chemistry to transform our molecules into products in seamless ways, helping to inform key decisions and guiding the development. And with that, I would like to give thanks to a number of collaborators who I have worked with, especially my former Lilly collaborators and Professor Sally Price over the years for the examples that I have shared with you. And I would finally like to thank you for your kind attention.
Okay, thank you very much, Susan, for the inspiring talk. I really enjoyed this presentation. Now, If you have questions to ask Susan, now is the time to ask you questions. You can also type your questions, or you can simply raise up your hand so that Susan will then take your question. I have a question here from Susan Bond. Susan Bond, would you like to come in and ask? Okay.
Hi, Susan. What an absolutely fantastic talk that covered so much. I have a range of questions for all different parts of your talk, but I started by typing this one quite early on. When you were talking about the olanzapine structure, and you were showing us the calculations and the order of stability that was obtained. And one of the things I found very interesting was the point that you made about higher order basis sets being used for those calculations to try and emulate real world situations, so real pictures that we're likely to see. And in that particular case, what you saw was that the order of stability was the same as simpler calculations. I just wondered whether there'd been enough similar studies done to perhaps indicate whether that's likely to always be the case, or are there examples where that order gets overturned?
So the one example, Susan, that I am aware of, because I think you're right, we're absolutely in the middle of accumulating that information for well-studied systems. The one example that I am familiar with is some work that Jonas Nyman and Graham Day did to be able to look at polymorph pairs. And I want to say that they had concluded that in about 10% of the systems that you could spot enantiotropic relationships between polymorph pairs, which would mean that the stability order would in fact invert.
Okay, well, thank you. That's something to keep an eye on then. Thank you very much indeed for a wonderful talk.
Thank you.
Oh, okay, thank you. There's a comment here from Simon. He says, "Brilliant talk. So great to have in our conference, industry, academia, and excellent interdisciplinary science." I just want to pose a question to you, Susan. To say, you talked about computational calculations or predictions, where you can actually predict drugs which with the low energy levels. So I just wanted to find out if this prediction can also give the probability of occurrence or crystallization of the different crystal forms that you are likely to get.
So the one thing about the crystal structure prediction methods is that they're obviously based on the thermodynamics and they're not looking at kinetics. I would point the audience to some recent work from Matteo Salvalaglio out of University College London. He's been partnering with Sally Price and others to use molecular dynamics to start heating up some of these crystal structures. And what he's found is that with metadynamics, you can actually melt a number of the structures by the time you get to ambient temperature. And other structures tend to coalesce to more thermodynamic ensembles. And this has been a really effective way to simplify the crystal energy landscapes that I've shared and give at least a window in terms of what crystal structures will survive. But in terms of actually nucleating and overcoming crystallization kinetics, this is where I see the absolute brilliance of data techniques based on, for example, the Cambridge Structure Database, because all of those structures somehow found a way to overcome kinetic barriers to crystal nucleation and growth. So can we start looking at the reasons why we are seeing certain structures and not seeing certain other structures? I think there's a tremendous amount of work that we can do in that space.
Okay, thank you very much. I think I just have one more question to ask. So from your experience, I know you have worked for quite some time in the pharmaceutical industry. So what sort of measures put in place to ensure that they don't they prevent polymorphism or changing of these crystal forms, because it's a huge loss to this pharmaceutical industry once a drug changes from one form to the other form. Do we have some sort of measures that these pharmaceutical industries put in place to avoid this polymorphism?
So I think the ability to avoid polymorphism isn't something we're ever going to be able to do. The only thing we can do, because it's out there, is hopefully get ourselves to where we can get stable forms so that we mitigate the risk of any transformations from that point on. And so this is why the industry has invested so heavily in experimentation, and now more recently, computational chemistry. All of this investment over the last couple decades has been to exactly that point, to eliminate that risk. Well, you can't eliminate it, but you can minimize it. And it's by doing, good experiments in a computer and good experiments in the laboratory, and combining the information to make the best possible decisions. And that's really where we are. And none of the techniques will be able to eliminate the risk.
Okay. So thank you once again, Susan, for the brilliant talk. I think you can engage with Susan and ask more questions. So this brings to the end of this plenary session. So from this, we now move to the breakout rooms. So in the breakout rooms, you should select the track that you want to attend, and that should be fine. So thank you everyone for attending this session. Let's move into our breakout rooms. Thank you.
Fantastic.
Does everyone
Thank
see
you.
where to select a breakout room? Look at the bottom of your screen, and there's a button that says breakout rooms.
Yes. Bottom of our screen.
And then when you click on that, a little window comes up, the list of the breakout rooms, and then there's a word that says join. It says join. You see, everyone see that?
Yep.
So, hi guys, I'm the next speaker in one of those tracks. I'm not getting this join option. It's not letting me join. There is no dialogue to join.
Did the list come up?
The list comes up, but there's no interaction with the list. I click on it and it doesn't do anything.
That's
What are you saying? You want to move into the breakout room?
Yeah.
So you click on the number. So you click on the number. It will ask you to join.
It's not giving me. I may need to leave Zoom and rejoin. It's not giving me any list of numbers.
Normally, the host
numbers,
should also be
words.
able to move you.
Yeah, well, I click on the words and it doesn't do anything. So yeah, if you could move me to the
okay. I'm going to move you right now.
Move me so I don't need to reconnect. Thank you very much, everybody.
Is anybody else having the same problem? Is everyone able to move? Who's not able to move to a new session, to a different session?
I'm still here, whatever here is.
Okay. Why is this option not working? I thought I moved you.
Right. I'm not. Yeah. So if you want me to leave and reconnect, I can do that.
African light source track. Yes. That's
right. Now I've been invited. Yeah.
Okay. Anybody else need help moving?
Hello, everyone. How's everyone doing? I wanted to, can you hear me?
Yes, yes, I can hear you.
Okay, great. Oh, hello, Trevor. How are you? I wanted to alert people that there are sessions in progress in the breakout rooms. So if you are interested in attending one of the sessions, on the lower part of your screen, you see something that says breakout rooms.
I see, that's how you get to them. Okay,
Yeah, you click on breakout rooms and then you go to the list. And there should be, at this point, there are several, but you can see the top 3 is where most people are. And what you do is put your cursor over the number. Do you see that? You put your cursor over the number and it will say join, and you click that to join whichever breakout room you want.
Okay, so let me try that. Okay, now what happens?
Okay, do you see African Light Source Track?
Yes, that's where I wanted to go. Join.
Okay, so put your cursors over the number 21.
So I should disappear if I tick yes.
Yes, you click join, and then that should take you to the room.
Okay, thanks, Sikosi. Thank you so
Oh, yeah,
much.
sure.
Yeah.
Okay. So others, if you want to go to one of the sessions, then click on the breakout rooms on the bottom. And then you will see a list of breakout rooms, and just put your cursor over that number, whichever, it tells you, for instance, if you are going to the African Light Source track, you have 22. Put your arrow, your cursor over that, and you will see the word join, and you click that. And it will take you to that particular, excuse me, that particular session. Okay, thank you.
Was everyone here able to get into a breakout room? Yes? No? Anybody having any troubles getting into a breakout room?
Yeah, I'm trying to. I think, should we first leave the room and then open it, or can
I
we
think
do
when
it
you're in
like
the main room now, so you just go to breakout room, and when you click on that,
that? Okay.
the rooms
Thank
come
you.
up, and then you see the little word join on to the right.
Thank you very much.
Okay. Anyone else need any help getting into a breakout room?
So people are now leaving the breakout rooms, joining. I know that the African Light Source just closed its session. So we should be started in a couple of minutes.
The other sessions closed just a bit before us, so I think you could.
Oh, we're all here?
Yeah.
Okay. Yeah, so why don't we get started? First of all, good day to everyone. So happy to see you. Is Jeremy Woodward, are you ready to go?
Yes. I'm ready.
Okay, great. So the first presentation in this session will be by Jeremy Woodward, who is from the ESRF, European Synchrotron Radiation Facility in Grenoble, France. Oh, I'm sorry. Oh, big mistake. I have it backwards.
That's not me.
No,
It's me.
From the University of Cape Town, right? So to discuss the cryo-electron microscopy that they have in process there, and I am fortunate a couple of years ago, Professor Trevor Sewell took me on a tour of his their fantastic facilities there. So with that, I will turn it over to Jeremy.
Great, thank you. I'm just going to start the presentation. No, this works. Can everybody see everything correctly?
Yes.
Yeah, okay, great. It's not in presenter mode or anything weird. On your side.
Yeah, we see your opening your title page, we see.
Excellent, thank you. Thank you very much. Yes, I'm from the electron microscope unit at the University of Cape Town. And over the last few years, I've been involved in cryo-electron microscopy. And I've been continuing in a tradition that was started by Trevor Sewell, who used to be director of the electron microscope unit. And so I'm going to talk about some of the progress that was kind of established by Trevor Sewell and how we are planning to move things forward and what is happening currently as well. Great. Okay. So the first point that I want to make is that the cryo-EM has become a really important technique. So at the very beginning of the current pandemic, a number of cryo-EM structures were solved of the COVID-19 or the SARS-CoV-2 spike protein, and a number of very important discoveries were made, which led to the development of vaccines, and they continue to be very important to highlight exactly what is happening with each of the mutations. So each of the different variants that are arising, exactly the mechanism of those and how they can be combated. One of the reasons behind cryo-EM's recent success is because of the resolution. So this is the current record resolution, which is at 1.2 angstroms. So this is normally a resolution that you would associate with cryo-EM. Most of the time, cryo-EM, at least X-ray crystallography, most of the time cryo-EM doesn't achieve this kind of resolution, but at the same time, most of the time, X-ray crystallography doesn't either. One of the other reasons that it's been very successful recently is because it's particularly good with large heterogeneous protein complexes and membrane-bound proteins, flexible proteins. Proteins don't have to be that pure, and very little material is needed. So while it is still true that the best samples are well-ordered, large proteins with high degrees of symmetry, it is also becoming increasingly possible to visualize smaller proteins. So this is a little bit of an older diagram. The current record is about 60 kilodaltons or so. for achieving something that is interpretable to atomic resolution. And if you look at the number of depositions, recent PDB depositions, you can see that cryo-EM is starting to catch up. This is not on a logarithmic scale or anything like that. This is directly comparable. And cryo-electron microscopy has come from being something that was basically at 0 to currently there are around about 10,000 depositions or so. And it's estimated that, well, it's perhaps optimistically estimated that in the next 6 years or so, cryo-electron microscopy will overtake X-ray crystallography as a structural biology technique. Okay, so at the University of Cape Town, we have been doing cryo-electron microscopy. Now, this was established by Trevor Sewell. The very first structure was solved in 2000. Well, it was before 2003. It was published in 2003. And so we're talking a relatively long history when you think about cryo-electron microscopy. But this period was a very important period because even though there were a relatively small number of structures solved over a very large period of time, so something like 20 years, and each structure was very difficult because the infrastructure wasn't in place, because a lot of scripting had to be done. There weren't very nice computer programs that could be used. One of the major reasons that there was this kind of difficulty is because the infrastructure for protein purification had to be established. The computing facilities had to be established. Even the electron microscopes had to be established. And so this was a period that can sort of be characterized as very few results and a lot of effort. So, so a very disproportionate effort to results ratio. And I think when I'm building facilities and when establishing a new technique, it's often like this, that there's a lot of work that needs to be done. Building up the community of people who can use the facility and establishing the resources that they can use, as well as, of course, the expertise. So what does all this look like? What are all the things that are necessary? Well, if we start over here in this green block at the top over here, these are the people who come to us. They've been doing DNA sequencing, they've been doing mass spec. These are people from different university departments. They come from all over the place. They've done a variety of experiments, and now they want to interpret what they've done structurally. And so the first thing that they do is they apply bioinformatics, modeling, molecular dynamics. They can use existing structures and do quantum mechanical simulation, and they can attempt to dock ligands and things like that. Now, very few people in Southern Africa are able to, off the bat, just use these techniques. And so we have had to build in resources and support for students to learn these techniques, and also to take established researchers through all of these methods. Once we've decided on an experiment, of course, we need to be able to do molecular biology, cloning. We then need resources for protein expression and purification. We need to optimize our samples. So this is the same with X-ray crystallography. Of course, X-ray crystallography just is a little arrow that comes off the side over here. But people often think that in terms of cryo-electron microscopy, there isn't much optimization because you don't need a crystal. But of course, there's significant optimization. Once we've seen that samples are good, we then need to ship the samples, because we can only do screening here. We can't get to very high resolutions. And so we need to borrow on the resources that other people have overseas. So this is large international infrastructures. And of course, to access these facilities, there's a barrier here. And that barrier is that you need to be accepted. So you need to demonstrate that your sample is of sufficient quality and that your scientific questions are important enough that really, really valuable microscope time is then given over to you. We have then increasingly needed to actually do high resolution data collection. When we first started using these facilities, there was a lot of kind of assistance given in terms of actually collecting the data. So very often you would just give your sample, you would sort of give the experimental parameters, and then data collection will be done for you. But now increasingly they just give you access to the microscope and to the software that one uses to actually collect the data, and data collection actually happens remotely. And then there's reconstruction, fitting and interpretation, and of course all the things that we saw at the top over there are now done on the new structures, and it goes along, and this is a sort of an iterative process. So of course, at this point over here, it's really just the beginning of the experiments, once you actually have a structure that's been fitted and interpreted. But there is significant infrastructure that needs to be put in place in order to get all of this right. And that in the South African context means a lot of student training, workshops, and a lot of equipment. So here you see the protein lab. This is really, really crucial because very few South African labs can produce protein of sufficient quality to do cryo-EM. And what I often tell people, and they can be quite surprised, is that when you're doing X-ray crystallography, the protein sample doesn't have to be of very, very high quality, because the very highest quality protein is then accepted into the crystal lattice. So you have a sort of a purification process that happens right there. Of course, if your protein is not pure enough and your protein is low quality, you don't form a crystal in the first place. But if you can, that crystallization process is actually a purification process. But with cryo-electron microscopy, the protein has to be perfect quality, because it's only 3 months down the line, or even 6 months down the line, that you actually figure out that your protein is low quality, because there is no crystal that forms. You just image the protein, and if it's low quality, you just don't get a good reconstruction at the end. But there's no way of knowing until the very end. And so we've increasingly become extremely paranoid about protein expression and purification. Then the samples are screened, and they go into the microscope, and screened again, and all of these processes are iterative. We make cryo-EM grids, which are then screened again, and once they are of sufficient quality, then they go overseas for data collection. So this in pink over here, in yellow, these are all of our resources that are in the electron microscope unit. Currently, the protein lab is under threat. The idea is to close it down, which is going to severely impact our work. But all of the rest of the resources are within the electron microscope unit at UCT. And then data collection over here in pink, that's international infrastructure. So student training is a crucial aspect of all of this. So we run workshops, and every year there are groups of honor students that go through the whole process. So they learn how to perform cryo-electron microscopy. We try to do outreach in terms of doing presentations in departments, so that investigators or supervisors can see the value in what we do and offer projects. But it is very, very difficult to get people to invest in a technology that is so intensive in terms of the input we need to put in. Samples really need to be good. Okay, so after all of this, what are the results that we can get? So in 2019, we published the first cryo-EM structure arising from Africa that could be interpreted to atomic resolution. So you can see the fitting of here is not great, but we could interpret what was happening. We got some great insights. So that was the very first. This is actually interesting. This is an unpublished project that was performed mainly by an honors student. And so during the honors degree, they had probably about 3 months of experimental time. And the microscope time was already applied for at Diamond Light Source. That was the international facility that we were using. And so the student could purify the protein according to known protocols, prepare the sample, send it, and actually even do preliminary analysis with a lot of support, of course. But during that process, a more or less untrained individual could come along and generate this structure. And so it really shows you that when the sample is right and when the infrastructure is in place and when the resources are available, a lot can be done and relatively easily. It also happened in 2019. There's one of the other structures. It's another filament. It's another helical structure. Various people were involved in this project, which happened over a number of years with several different data sets. This was being driven by Trevor Sewell.
Okay, could everybody mute except the speaker?
Sorry. I'll continue. Yeah, so this was another project, and eventually it got to a resolution that was good and it was interpretable. In some cases, like for instance, this is an HPV project. So this is HPV 16. The idea here is that in order to become infective, HPV undergoes a conformation change. And this has been demonstrated previously with antibodies, but it had never been visualized. And so we, for the first time, visualized cleaved HPV, but the resolution was quite poor. It was at 4.6 angstroms, and you can see some regions over here. This actually looks far worse than 4.6 angstroms. So in this particular region, it doesn't look good at all. You can just see the positions of alpha helices. But we could still get insights because the overall conformation change was still visible. And so the entire chain kind of twisted. And you can see the blue chain versus the green. That's cleaved is blue, so it's furin plus, and green is the uncleaved structure. And so there's this relatively large scale conformation change, and we could demonstrate that it had an effect on the ligand. So the ligand binding improved when it was expected to. So even when high resolution cannot be attained for whatever reason, in this case, I think it was because this particular protein or this particular virus was expressed and purified outside of our lab. And I think the protein sample was a little bit poor. So this had actually been frozen previously, which is never a good idea. This is another project that we did the data collection for this. We did that in October 2019, and it's now, let's say, October 2021, which is 2 years later. And finally, the structures are completed and interpreted. So this was a particularly long processing problem. And the reason for that is because this particular protein ended up in 2 different forms. This is a monomer, has 2 domains. And it also formed this kind of flexible dimeric form with 2 of these monomers with 2 dimers, the dimers associated. There was flexibility about this region here, this neck region. So the 2 domains could flex relative to one another. And it was also glycosylated. So this is a heavily glycosylated protein. And so that also introduces a lot of flexibility. And so all of these degrees of freedom mean that in terms of processing, one has to eliminate all of the other forms. And so one of the successful strategies was to mask out one of these domains and just reconstruct that, and then another domain and reconstruct that, and then just the interface between these two domains here. The top part over here is obviously defined by these flexible linkers, and so that part at the top is quite flexible, but very good resolution was attained for this interaction. Here's another reconstruction. This is of a nanocompartment from Mycobacterium tuberculosis. This is exported from Mycobacterium tuberculosis under conditions of oxidative stress. And when you knock it out, the bacillus becomes way more susceptible to oxidative stress. And so recent experiments, sort of functional experiments, have gone very far in determining the function of this particular system. And this is a reconstruction that is now, you can see that we're improving. This is a 2.2 angstrom resolution. And so we can really define exactly what is happening here. We get some wonderful insights because the substrate has to pass through this little channel here. This is at the 5-fold axis of this icosahedral kind of hollow sphere. The substrate passes through here, and there's a selectivity that happens at this point, and of course you can define that if you can see it at high enough resolution. And here's the final one that I want to show. It's a further 2 of these helical arrays. And what I want to demonstrate here are all of the known enzyme types of this particular type. And you can see this very interesting relationship between the overall helical twist. So that's this macromolecular helix. I'm not talking about an alpha helix or something like that. I'm actually talking about these protein subunits associated and forming along a filament. The helical twist of that filament is very closely correlated to the substrate size. And the reason behind that is because the binding pocket is at this little red circle here. And as the whole filament kind of twists around, we imagined that it would kind of compress the binding pocket. But the new information that we have is actually that it's the opposite, that the binding pocket, by being compressed, exerts an outward force. And that force then changes the helical twist. And so when we could see this effect at low resolution, we hypothesized the particular mechanism behind this. But now that we have high enough resolution, we can actually see the real mechanism. And it's almost the opposite of what we thought. And so all of these results come from, so the data was collected in 2019. In the one case, it was in 2018. But most of this data was collected in 2019 and 2020. And so in a relatively short period of time, we've shown that we can actually get pretty good results. That's not to say that we've forgotten those older results in negative stain, so we still do a lot of negative stain. This is spike over here. This was expressed in plants. It's a very interesting project because they ended up with very high quality spike, which could be reconstructed quite easily, but at least at low resolution, after being expressed in plants. Whereas mammalian expressed material is very sensitive. This is a kind of a nano cage. This is a designed protein, and various other projects. So these are always occurring. Okay. So currently, in terms of the management of these microscopes, these are high resolution cryo-electron microscopes. They're managed almost like a beamline. So it's a sort of this particular cryo-electron microscope is actually at diamond light source, so it actually sits on the synchrotron floor, and the management and the access and the administration and maintenance all occurs as though it were a synchrotron beamline. And so we've tried to replicate this. So this kind of new concept for which we have funding for now is a Southern African cryo-EM network. And the purpose behind it is to increase cryo capacity in the region. There are two other universities in South Africa, the University of Pretoria and the University of KwaZulu-Natal, that have some equipment for cryo-EM, but they lack expertise. And what we're doing is we're going to implement a test case. So we're going to test the model of free access, as long as it's non-proprietary research, with access being determined only by a peer-reviewed system. And it's going to be based, so this is going to occur over the next 3 years. We have funding for the next 3 years. We're going to assess projects on the basis of their science case, as well as their sample quality. And we're even going to assess people's samples quality. So people will be able to send samples to us, and we're going to be able to assess them. We're going to employ somebody exclusively to work on this project. And we have funding for all of the scientific expenses, the same as the synchrotron. And a limited number of people will receive travel grants to travel to the University of Cape Town and to go through the process of optimizing their sample. So these are people who are going to be outside of the Western Cape. People from within the Western Cape must stay at their own houses, but we're going to pay for all of their scientific expenses. But those from outside, we're going to pay for travel and living expenses. The applications are not available yet, and I can't announce who the funder is yet, but they will be available on this website for anybody who's interested. So it's just emu.uct.ac.za. And here's really an illustration of what we want to do. These borders over here are not. We're not saying anybody who lives in the blue is not welcome. We're just saying that we are focusing on Southern Africa, and so we will accept applications from anybody, and it obviously depends on travel funding and things like that, whether or not they get accepted. But in terms of individual projects receiving a yes or a no, that's only going to be based on the science case and the sample quality. So there is Pretoria and there is Durban, where the University of KwaZulu-Natal is. So these two facilities over here, we're going to ensure that they can do cryo-electron microscopy screening, and then samples will be sent to the University of Cape Town, where we can do higher level screening and then of course shipping to international facilities. Thank you very much. Yeah, that's all I want to say. I haven't acknowledged the new sponsor, but I can't release that information yet. But the work up until this point was sponsored by the GCRF through START, which is the Synchrotron Techniques for African Research and Technology, and the cryo-EM work that I showed. It's not the only cryo-EM work that has been done, but the cryo-EM work that I've shown is from Diamond Light Source. Thank you. That's really great.
Thank you, Jeremy, for such an excellent presentation. We do have time for a couple of questions. Jeremy, could you stop sharing your screen so it will be easier for me to see hands that are raised? Are there any questions at all? Just raise your hand if you have a question. Okay, I don't see any. Okay, with that, we will move to the next speaker. Thanks again, Jeremy. So the next speaker is let me get this right. I don't want to say a mistake. The next speaker is Gianluca Santoni, who is from the ESRF, and his talk is extremely timely since we're all facing this whole coronavirus pandemic. So at this point, then I would turn it over to Gianluca, who will discuss the coronavirus structural task force. Okay.
Hello, everyone. Can you hear me?
Yes.
Great.
Yes.
Okay, so it's a pleasure for me to be here to present the work of the Coronavirus Structural Task Force. As you will see, it's a work of many people that will be acknowledged later on. But so just to start with, first, let's focus on the coronavirus before focusing on the task force itself. So you might by now be familiar with the coronavirus and how it looks like. Is this envelope formed basically by a membrane and different structural proteins? Each of these proteins, obviously, we can zoom in at different level of details. So this is the first image that we can get out of a spike, for example. But if we start zooming in more and more, we can see that there's more to analyze. We can guess the structure of the protein inside there. If we zoom even more, we can focus on the different domain of the spike, in particular the external part, the subunit 2 that interacts with the human receptors. If we want to go even in much more details, this is those structures as already presented before were done mainly by cryo-EM, but we can focus on the individual details, of course, of this protein, and you can see here a glycosylation site, all the atoms. And if we move forward, actually, we can realize how big the challenge of determining a structure of this kind of problem is. Here you see all the atoms inside the spike, which is quite a big number of them, thousands and thousands, of course. But the coronavirus is not only the spike, there's way more. First, we have the virion, the picture I was showing before, so the structural protein, the membrane, and the RNA enveloped it inside. The virion enters the cell via using the spike that interacts with the receptor binders, with the receptors on the membrane itself. And as I said before, there are 4 structural proteins in the coronavirus. Once in the cell, the virus releases its RNA, and this RNA is decoded into the cell into a polypeptide. This polypeptide is actually contains, more precisely, 16 non-structural proteins, whose role is to hijack all the cellular machinery in order to replicate the virus. They're still not clear how everything happens inside the cell itself, but main task is to suppress the immune response of the organism to favorize, let's say, the replication of the virus. And then different proteins take care of different tasks. For example, the RNA-dependent RNA polymerase that we will see later multiplies the copies of the viral RNA inside the cell. The virus is then somehow, it's not very clear, released from the endoplasmic reticulum into the cell itself and meets its own, it's again not very clear, but the idea is that the virus reforms itself, the RNA is protected by new copies of the nucleocapsid protein, and then the new virions are released inside the organism. And there's not only the structural protein, we have another 8 proteins which are encoded by the viral RNA, but for which the function is rather unknown yet. They're usually called accessory proteins because we don't have no real clue what's their role in the life cycle of the virus. The different structural proteins and non-structural actually are ideal drug targets for drug screening. For example, here, the classic example of the RNA polymerase, where we can see an inhibitor, is bound into the active site. Or the spike, which is the main target for vaccines, as again has been presented very carefully. And it's important again to know this atomic structure of the spike because variants of the virus are mainly classified by the structure and the sequence of the spikes. Where do all these structures come from? I have a very annoying bar at the top of my screen. Mainly from X-ray crystallography, NMR, and cryo-EM. Those numbers date from a couple of months back. At the time, we had the 1116 cryo-EM X-ray crystallography structure, 32 NMR structure, mainly the envelope protein, but a few others, and 421 cryo-EM structure, including the spike, RNA polymerase, etc. Problem is that this is really okay. Well, now it's better. Errors in structures are normal because, as we've seen before, building the structure is quite tricky. Okay, yeah, it's a complex problem. The fit between the data and the model is not always perfect, and it will never be because the data themselves are not perfect and not ideal, and they don't represent a single model of the protein. Complete automation of the whole structural biology pipeline is not yet possible. A lot of people are working on that. And one thing which is important is that models are mostly built manually for a new protein or built based on existing models which were built manually before. We'll see this in a later example. Expertise in many different aspects is needed. That was presented just before me. Solving a cryo-EM structure is not just a matter of getting the micrographs ready. It's a matter of having the pure protein well spread, the uniform distribution of protein states, etc. One of the things that is needed is software. We tend to forget this sometimes, but without all the software that are kindly available in the world today for structural biologists, we will not be this advanced in many fields. Errors are normally common, and by now I mean now 18 months ago, but we had the pandemic, people dying around, public pressure on researchers, etc. Scientists were cut off their regular, let's say, ways of interaction. They couldn't access their lab as before. And in the meantime, so we had limited possibility to work as we are used to, but we are also required to provide all the information needed to move forward in the fight against the virus. So with a group of friends, started by Andrea Thorn, who is the leader of the task force actually, we said, how can we help? And the task force came out as a collaboration of spontaneous willingness of people that, let's say, we thought we could do something as method developers in structural biology. This is a picture of the task force. We are just missing one very important person, which is Jane Richardson from the pictures. So the group, as you see, is built by a lot of students, a few senior scientists, and many young people, I would say. But in the end, we managed to do quite a lot of work to get out. So what did we do exactly? As I said, we were mostly method developers in different structural biology technique, mainly software. So we decided at the beginning, the idea was we will evaluate for a couple of weeks the few structures from SARS that were existing at the time and just move forward from that. We realized very quickly that structure from SARS-CoV-2 were flowing in way faster than we expected. And so the project from a 3-week thing became an 18 months ongoing collaboration. For example, we had to give up the study of the, let's say, the evaluation of the human interaction partners because that was piling up a bit too much on our, let's say, workload. What we did, the evaluation was done in different levels. We were reprocessing the existing data, remodeling the structures where needed, evaluating their quality with different tools that I will present on, and did some scientific dissemination and public outreach. What we do, so first we built an automated pipeline that fetches the PDB every week, compares each single sequence with the known sequence of the different proteins from SARS-CoV-2 and SARS virus, classify them in different folders, and then we perform some automated calculations. We check if raw data are available, and after that, we download the structures, and we perform an automated validation. The automatic evaluation performs a series of different software that can be run on each structure, and for which we tend to gather together all the results in order to provide to the final users a, let's say, more complete picture than the simple sliders that you can find on the PDB website. Then, when something is really, really wrong, we have a group of people who can reanalyze and reprocess the structures manually. We have rebuilt different models. We have sometimes reprocess the raw data when available from the users and from the depositors themselves. And all these, let's say, allowed us sometimes to really improve the existing structures or to point out some issues with them. All our work is then disseminated via GitHub. We have a repository which is weekly updated with all the automation and the new structures. And then we do outreach on Twitter. We have 3D models of the virus available. We will see that later on. And most important, we contacted authors of the original entries to have them edited on the PDB. So people can in general not, let's say, nobody but the original depositor can change a structure on the PDB. And so we took care that at least some which had the most striking, let's say, issues that we found would improve that. And another thing that we did a lot of care, we took a lot of care of, was to ask authors when important to deposit somewhere the raw data for their experiments as well. We did explanation on Proteopedia and through our own page, and we are writing some reviews. So some statistics from the X-ray crystallography, as we are speaking about light sources here, maybe is the most interesting to check. Interestingly enough, the structures came from basically all the synchrotron around the world. Diamond is the clear champion in numbers, in particular due to the XChem studies on MPRO and I think they have another 2 ongoing. Distribution of software is quite spread out. XDS is obviously the most used, as one would expect. Surprisingly enough, Phoenix is used almost twice as much as RefMac for refinement. And there's quite a lot of people who use Buster in their pipelines. This is the resolution, MX resolution versus cryo-EM. Obviously, you see the mean of the 2 is quite, let's say the mean of cryo-EM is higher than the mean of protein crystallography. This is for 2 reasons. Most of the structure as in number from MX come from the MPRO, for which the crystals diffract extremely well. So this tends to shift, obviously, the distribution towards the left for MX. And for cryo-EM, obviously, we have the instrument limitation and the sample limitation that were described before. Interestingly enough, on average, the structure look okay. Let's say if you check the R3, it seems quite fine. Nothing to, let's say, nothing really bad on the overall picture of the structures that are released. One thing that we present in our database is the comparison of all the structure. For example, we calculate the RMSD between of all the possible pairs of structure per protein. We show here the endornase example because there are not many structures, so it's quite easy to see that 7K9P, for example, is quite different from all the other structures deposited. If you look in our repository, you can find other useful information where we got the sequence, a link to the blog post that exists, the possible glycosylation site calculated via web server, the information on the proteome, etc. So all those information allow people to have a more clear understanding of what's behind the simple PDB coordinates that one can download. And this is what you get when you go to our website. So there's an article explaining the function of the protein. And we had made for all the non-structural protein by now. Okay. The important thing is what is that we need, so the moral of the story is that we need to get the best out of the data. Once the data is measured, the data is there. The only thing that can be improved is data processing on a side, and so on a side is the data processing, and the other obviously is the model building. If the data is available, we can try to reprocess and redo our best with both the model and the data. Otherwise, we will try only with the available process data and the model building itself. Looking into all those structures, we found many different modeling problems. Sometimes it's quite striking, like this structure here, where the Ramachandran plot is quite a mess. Sometimes you need really to go into the detail, because you see, for example, here, this zone around an inhibitor where the electron density map and the model built inside don't really fit to each other. Other weird things like this for cysteine pointing towards something. This was edited obviously later on by the addition of a metal ion in the middle. Sometimes there's strange coordination that can be solved by just flipping a residue away. Here again, zinc are quite bad thing around. But yeah, for example, here the zinc would have made 5 bonds, which is clearly not the best. Trans residues versus cis residues are quite appearing. Flipping chirality in glycans are also, let's say, often observed. This was as easy as flipping the first bond. And now we will go on to, let's say, an example of different things that we found in an early structure. So you all remember the story of remdesivir, who came out was what, like May 2020, as an inhibitor for the RNA-dependent RNA polymerase. Remdesivir is basically an adenosine analog. The idea is that this will bind covalently to the RNA polymerase and stop the replication of RNA inside the structure. As nicely depicted by our PhD student, Alex Payne, basically remdesivir acts as a Trojan horse for the RNA-dependent polymerase. Will stop the replication of the RNA. This is another thing that I didn't mention before that we do to our structure. This, for example, the remdesivir-bound RNA polymerase, is that we can apply a neural network to predict the secondary structure of the problem from EM maps. This is particularly useful for model to be built de novo, but it's also interesting here because we can see that we use this actually to compare our prediction with the model built by the users. You see, we use the same color scheme. So the RNA is in gold, helices are in red, and beta sheets are in blue. You see the resemblance is quite good, which can be read in the two senses. Either our software does a good job, or the depositors did a good job as well in building the model inside this particular map. And you can see here in the middle is where remdesivir binds. You see it's basically at the entrance of the actually at the exit of the active site. So how does this work? This is again the electron density map for remdesivir. You have the two RNA strands, you can see them in the middle there. And yeah, so one is the original, which is the green one, and the gold orange one is the copy. As you see, remdesivir takes the place of a base of an adenosine, which means the copy cannot go on anymore. So it will stop basically the copying again of RNA. And as you see, there are in the deposited structure, we can find 3 magnesium atoms inside the structure, which were put there by the depositors. But yeah, it's quite difficult to see here. But basically, as you can see, the density has no support whatsoever for those magnesium atoms. Sorry, my thing got stuck. Yeah, so yeah, there it is. So we asked the depositors that maybe it was a good idea to remove them, and the structure was updated later on to remove those. And while building this structure, actually, we realized that there was another issue with it. So around the C-terminus, there is an helix, which in the first RNA polymerase structure from SARS, it was extremely badly resolved. And so the depositors originally just deposited with what they guessed, let's say, will be the nice sequence. But when you look at the first deposited structure for the RNA polymerase for SARS-CoV-2, you see that there is quite a bad fit in all this region. And this was due to the fact that there was actually a 10 residue shift in all the region that we were now able with, let's say, more modern tools to correct against the more recent data for the RNA-dependent RNA polymerase. And now the fit is way better. And this has been since also solved, and this part of the publication put in link. So we had a lot of partners for this project. It's important to know before we go on that actually this is a private communication from the PDB, but about 90 to 95% of all the downloads from the PDB don't come from structural biology people. They come either for biologists or for molecular modelists. So people who don't really, let's see, whose main expertise is not the model building, so could, let's see, neglect the fact that there can be errors in the structure which is deposited. And yeah, this is just a very long list of our partners. So once we had all this information, we tried also to do some more outreach on how the virus could look like. This is a first picture we got, but then we realized, for example, that the spike was a bit out of scale, that the number was not correct. The distribution, when you look at models for the coronavirus, the distribution of spikes is usually quite uniform on the surface. So we work with all these to try to build a better model. This we also updated because more recent study actually found out that there are way less copies of spike than what was thought at the time. So when we built this, we thought that there were about 100 copies of the spike around the virus, and the number now set, let's say the consensus is more settled between 30 and 40 maybe. So we have a new model coming out with that. Because with this model, actually, we release the coordinates. You see the difference between the two, of course. And before we go to the coordinates, when we look inside, we can also see one of the hypotheses on which the RNA is actually replicated and folded inside the protein enveloped by the nucleocapsid protein. With this model, the interesting thing is that it can be printed. We have printed a few copies. Sadly, I don't have one here, but those are very useful because first you can download, I don't have the link here, but you can download them and you can print it by yourself. It's quite a long process, but you can then paint it, and they're quite a useful tool to understand actually the shape and proportions of the virus itself. And here we have an antibody bound on the spike, where we made a model where the antibodies can actually bind using magnets, and that's particularly useful again for outreach activities. One copy of the model was part of an exhibition in Germany in a museum about the pandemic. So go towards my conclusion, and actually what's the biggest meaning of this, apart from finding obviously problems inside coordinates, which is obviously important. Important is the fact that the pandemic actually changed our way to do science. As I said, the task force was an effort of up to, at some moment, we reached almost 30 people. We were spanning 9 time zones, and we were trying to work together to gather all this information and to clean it up and to make some reasoning out of it. This is a regular working day. There are a few people in Hamburg, and then most of us around on Zoom. Interestingly enough, this is another thing that came from the pandemic. I think that's probably the first time the structural biology was this much under the lights. Andrea appeared a few times on national TV in Germany. We had a few journal articles. So all of this had quite a lot of impact on society. To the point that we also had our first fan art. This is just a girl who read about us on, actually shared the podcast when we were talking about our project, and she drew a picture of all of us, which is super nice, I think. But yeah, let's probably can go to the summary where I do my conclusion. So first, to understand the virus and this life cycle, we need to understand the biology. I think this is clear for everyone, and understanding the biology helps in designing and targeting the therapeutics for the virus. What we did is, again, evaluate manually and automatically all the structures. And finally, this was quite a struggle, but we managed to have a publication for this. You can download it using the QR code. I will leave it for a second on the slide. This also exists as a preprint, but I think it should be in open access now on NSMB, so there's no problem with that. But the important points are these. First, the structural biology is difficult, despite the fact that you can hear that now the folding problem is solved. This is clearly not the case. And there's a lot of things that we can learn from our finding, maybe. We should change our attitude probably towards 2 things. One is toward error, and 2 toward the deposit methods. The deposit method is not the, let's say, the truth carved in stone, but rather is the best interpretation that the author could do of the data that he had with the instruments in his mind, in his hand. We had this question many times: should this serve as a model for other projects? So, and this is the reflection I wanted to make, because as a, let's say, junior scientist, and I think there's a lot in the audience, we know that we are always going towards the publication record, etc. But it's also true that we should aim towards improving and giving back to society. So this kind of completely open method where, well, in the end, we managed to come out with a publication, but it was, one, a big struggle to have it accepted, honestly. And 2, it was not our main focus. Our main focus, again, was the data, as we are. It's really tiring. It was really difficult to gather some funding. Luckily, the German Ministry for Research gave us some money and allowed Andrea to use some money she already had for the project. This is, again, not really easy. Another question that came out, again, is how senior do you really need to be in order to be a leader of a project, but this is obviously mostly for Andrea, who was the leader of the task force, and how can complete your, let's say, as open as we can science project compete with in the, let's say, current scientific career market. And the last question, probably this is more for reflections for everyone, is that what will change in science after the pandemic? Will the scientists be viewed differently by the public? Probably not, but we will see. And with this, I'm at my last slide. So need to thank first Thomas, I will never be able to pronounce his family name, who is a graphic illustrator, and he made all the nice pictures that you've seen all through the presentation, all the task force members and collaboration partners, in particular who we called our scientific fairy godmother, Arwen Pearson. And I will personally thank also the ESRF Structural Biology Group for allowing me to participate in this. And obviously thank the organizer for inviting us, and thank you all for your attention. And obviously you can find us on social networks as well. We have a channel on YouTube, and you can find me and Andrea on Twitter.
Fantastic. Thank you so much. I learned a lot from your presentation. Are there questions? Please raise your hand if there are questions. Let's see, let me do a quick scan here. I don't see a hand anywhere. I do have a question. You know, you're at the ESRF, which is one of the world's leading advanced light source facilities, and you know all about the cryo-EM. And the question is, you have these two methods of deciphering proteins. Of course, you have the neutron scattering that was mentioned earlier, but which size proteins are better deciphered at a light source versus cryo-EM? Can you touched upon a little bit when you talked about the resolution of the two competing facilities. Well, not competing, but complementary facilities. So if you could say a word about that.
Yeah, so first, I think the figure was also in previous presentation, where I think it was correctly mentioned that the lower limit for cryo-EM sits somewhere around the 60 kilodalton. So for cryo-EM is just the size of the object that defines the achievable, not only the size, but let's say the size of the object will be the main enemy against the ability to solve a structure with that. For protein crystals, for crystallography is a bit the opposite, in the sense that in general, I would say smaller proteins tend to crystallize a bit better than bigger ones. There's less, I would say the packing tends to be more efficient. I think, someone can correct me if I'm wrong, but if you go on the PDB, you can clearly see a trend, almost a straight line of dependence between protein weight, molecular weight of the protein, and achieved resolution. So the smaller the protein, the better for crystallography, because again, it can pack more tightly and more with more order. So yeah, again, for example, just to give an idea, bigger complexes in general are really difficult to solve by protein crystallography, because they have this additional degree of freedom, which is you don't only have to sit, it's not showing on the camera window. So you don't only have to see two copies of a protein nearby, but if you try to sit in a crystal in an ordered way, two copies of a complex, this complex has also its internal flexibility that will break the symmetry of the crystal, basically.
I see. Thank you so much. I see Silvio Nesti from Electra. Maybe you can also comment on that. Go ahead.
Well, it was a very interesting talk, and I think it raised a number of questions for the whole structural biology community. I mean, I don't quite agree that the majority of the structures are actually deposited by biologists. I think there is also a bias. I tend to put the names of my collaborators, but I do think that we are in a moment in which, you know, all of the students in my lab spend most of their time making proteins, doing biochemistry, and even when they are lucky enough to get a crystal, that's the end of their PhD. So the training, there is a problem with training in crystallography. And I start wondering, should we leave this to expert, to you, to Tassos Perrakis, I mean, I'm not really sure which is the answer right now, but I think we are at a serious, somehow.
Yeah, that's a really fair point, Silvia. I completely agree with you. So what I meant is not really that it's only biologists that deposit structure, but rather it's not biologists that download the structure afterwards, which is even more important to point. To answer your question, on a side, I would say no, in the sense that obviously the few, as you label, experts in the world cannot stand up with the 300 something structures deposited on the PDB each week, of course. But that's not the main problem. The main problem is that those structures were solved with a goal, and the goal was to answer some biological questions. I can tell you if some data is a problem, because I sit at the synchrotron most of my time, so I can see that quite easily. But I cannot know each single protein what it's supposed to do, why a bond can look strange, etc. So collaboration with, let's say, experts on data processing are a good thing, but the interpretation has to be done, obviously, interpretation and also the model building, because the model comes, let's say, follows also the complementary experiments, and that has to be done by the people who produce the protein, because they know what they're looking for.
Mm-hmm. Thank you. Let's see, the last question is from Abidemi Kapo. Please, sir. You're muted.
All right, can you hear me?
Yes.
All right, thank you. Yeah, I've listened to Gianluca's argument and also Sylvia. I just want to say that from the side of structural biology, what we have, if you have a student on a project, structural biology project, like Sylvia said, is you work on the protein, which is always, like the rate determining step, and it takes a while to get a pure homogeneous protein. And after that, you get crystals. So the time is not really there for most students to actually finish up the whole pipeline of studying the structure and then answering the biological questions that, that comes from that. And that is where, postdoctoral training comes from. And I am much persuaded that we can get a lot of our students into postdoctoral training so that they actually can learn and complete the whole pipeline. I listened to the talk of Jeremy this afternoon, and you will see that a lot of the work that Jeremy has presented was actually work that started from Trevor Sewell's lab, during his PhD, all the way through his postdoc, and now, so it takes a while. And the opportunities, especially from the African perspective, is actually not really there. And getting the right collaborators to work with as well, it's something that as African, biologists are grappling with. So it's actually a good thing that we've got experts like Gianluca, Sylvia. I see Trevor before. I don't know if he's still around. So, and Trevor can actually, confirm what I'm saying. So the will, I mean, especially the political will, to push, this type of research, to the next level is also needed, from our African government. And all in all, we can all work together to make sure that everybody has these skills. And then we as Africans can say, okay, yes, we are really doing, something. So I think that's just my small contribution. But I believe that postdoctoral training or post-PhD training is actually needed so that students can learn the whole, pipeline of, from protein all the way to structure and then the function. Thank you very much.
That's an excellent comment. We will have to definitely keep that in mind. Thank you. Okay, so I think this brings us to the conclusion of this plenary. And again, I would like to thank the two presenters. We've learned a lot from their presentations, and I hope that we are all energized to go out and conquer some of these macromolecules. Silvina, do you have something that you were really pressing to say? You're muted, though.
It was.
Oh no, I saw your hand go up. I thought you were just clapping. Okay, great. Thank you, everyone. So we'll take a break now. We're running a little behind, but in 5 minutes, the next sessions will start. So I suspect they will start maybe in 10 minutes. So at this point, thank you, everyone, and we will see you shortly. Thank you.
Thank you.
Hi everyone. Can you hear me?
Yes, we can.
Okay.
Yes, we can hear you.
The next, just to inform you that the next session, the minister forums will be held on the AfLS breakout room.
I'm sorry, could you repeat that again?
Okay, the next session, the Ministers Forum, will be held in the AfLS breakout room. So if you want to attend this session, you need to go to the AfLS breakout room now.
Thank you.
You're welcome.
Lawrence? Lawrence?
I don't think Lawrence is here yet, Kotze.
Hi, Trevor, how are you, sir?
I'm fine, thank you. How are you?
Lovely to see you, Trevor.
Nice to see you. Well, you haven't seen me yet. I can put my... I'm in full relaxed mode here, sitting in my study.
Okay.
Folks, I'm just going to chip in again briefly to let you know that there's no session happening in this main room now. Both of the sessions that are happening now are happening in breakout rooms. So if you want to join the AfLS Minister session, you need to join the AfLS breakout room. And the African Political Society is also in a breakout room.
That's where we were, Delia, until Lawrence told us to come into the main room. Lawrence transported us from the breakout room into this room.
I thought we were going to caucus a little bit before we got started, but the people who were supposed to caucus with are not here yet.
Lawrence.
Oh, there you go. Everybody seems to be here now, so I will turn my face off.
Someone, you're muted.
Simon.
Yes, we should bring everyone to this room. Is that it, folks?
True enough, but I do not see
I'm going to go scavenge people from the other rooms.
Okay.
Bye-bye.
But I think the people who I'm looking for are not even in any of the rooms.
Lawrence, the South African minister is being represented by Dan du Toit, who is in the room.
Okay. I'm looking for the commissioner from the AU, who I do not see. Let me try to make a phone call.
Hello.
Hello.
Hello, hi.
Hi, I'm
Yeah, I'm Professor Koubea from Côte d'Ivoire.
Yeah, me is Ngu Loic from Cameroon.
Yeah, I experienced some difficulties for my connection, and I just succeed to connect. So I joined the meeting now, and I'm from Abidjan, Côte d'Ivoire, and I represent the Ministry of Higher Education.
So are you going to be representing the minister himself as a?
Yeah.
Oh, so the minister will not be joining?
Yeah, the minister himself is on trip. He's not, he went for the mission in France, and he is not present actually. So I represent him.
Give me the name again.
My name is Koubea, K-O-B-E-A.
Koubea.
Koubea. And I'm a director of the Cabinet of Ministry of Higher Education.
Director of Cabinet.
Yeah, of the Ministry of Higher Education.
Doctor, Professor, Mr.
Professor.
Sorry, sir.
Yeah.
By demoting you to doctor.
Yeah. So nice to meet you.
Khotso Mukhele is my name. I'll introduce myself more fully.
Yeah.
So we still await the Nigerian minister or his representative.
Okay. Okay.
And the commissioner, who I do not see still.
And the commissioner, AU commissioner.
Let me see what's happening here.
Hello, Dan. Dan, can you unmute?
Yes, Dr. Mukele.
You must never decide where you think you'll meet me or not meet me. You'll meet me in all sorts of funny corners. But if the conversation is Pan-African, almost rest assured that you'll find me there.
Very good.
Are you well done?
I'm very well, yes.
Were you in Paris last week for UNESCO?
No, it's actually this week, the Science Commission, but our minister is there.
Okay, okay.
So last week was the plenary of the General Conference, but today was actually the adoption of the Open Science recommendation this morning.
And it's gone through?
Yeah, it's gone through.
Okay.
Just, Dr. Mukele, I'm not sure if people are aware, but it looks like we're being live streamed on YouTube. So even if we make small talk amongst ourselves,
okay,
between it, because on my screen it says live on YouTube. So
yeah, it says it on my screen as well. I hadn't picked it up then. Sorry about that.
no, I'm not worried. Nothing untoward has happened, but I think just for everyone to be aware.
I have been to the other rooms and scavenged people, and I think we can regard this as our meeting. And Professor Kotomokele, you are free to start it.
Can I get from Lawrence that he concurs with you? Lawrence, are you still there?
Yeah, I don't see the commissioner or any of the commissioner's people.
I suppose you will alert me, Lawrence, as soon as either the commissioner or the Nigerian minister or his or her representative join us. You'll let me know.
Yes, sir.
Okay, good afternoon, ladies and gentlemen.
Hello.
Hello.
Yeah, good afternoon.
Okay, I can see that we have 62 participants in this session. I'm sure that others may join us along the way. As Dr. Joy just alerted me and all of us, we are being live streamed on YouTube. And so let's all be cognizant of that. My name is Khotso Mokhele, and I'm a member of the African Light Source board, and for now serving as chair. And I'll be moderating the session. When the session was initially planned, we had possibility of ministerial participation from Cameroon, Rwanda, Egypt, South Africa, Cote d'Ivoire, Nigeria, and the AU as well. I'm informed by Simon that Cornell that the minister from Benin joined the session earlier and articulated her country's position on the African Light Source in that session. I have been informed that the minister, the South African minister, Dr. Blade Nzimande, is currently in Paris at UNESCO because the Science Commission of UNESCO meets this week. This being the General Conference of UNESCO, which started last week. And we've also been informed that the Professor Adama Diawara of Cote d'Ivoire is also en route to Paris, probably going to UNESCO as well. But Professor Kobia, who is the director of the cabinet in the Ministry of Higher Education, will represent the minister in this forum. Dan Dutoy is the Deputy Director General in the Department of Science and Innovation of South Africa, who will represent Minister Blade Nzimande. In the event that we're joined by the Nigerian minister or the High Commissioner, AU High Commissioner, you will be informed. But we will kick it off with the two ministerial representations from South Africa and Cote d'Ivoire. The participants in this session would be aware of the enormous amount of work that has taken place as a bottoms-up work to develop a community that recognizes the value of the, as Simon, you've raised your hand?
I just wanted to inform you in the chat we learned that the Nigerian minister will join in 30 minutes.
Okay. So the moderator is going to have to moderate this very fluidly, and which we shall do the best we can. Simon, thank you very much. You can lower your hand now. So a lot of work has taken place over the last 10 years plus. I recall when I was still the president of the National Research Foundation, talking to Simon and Sakazi and others about the possibility of this continent building its own African light source. And part of the inspiration for believing that a project of this nature can materialize on the African continent came out of the ability of Africa to build two major telescopes, one being the optical telescope built in Sutherland, South Africa, and the radio telescope called the optical is the Southern African Large Telescope, and the radio telescope, the Square Kilometer Array, also being built in the Karoo in South Africa. These two mega projects, so SALT was a mega project at the point of its building, but SKA has come and dwarfed SALT and become a true mega project on the African continent. And even though this SALT is an instrument built in South Africa, SALT is considered as a Pan-African instrument. The SKA, on the other hand, the core of the array may be built in the Karoo in South Africa, but there's elements of the Karoo that will radiate out of South Africa into many African countries. There's already a dish in Ghana, and so it is also a mega Pan-African telescope. And Dan Dutoy is here. I'm sure he can talk to us more about these two projects. So my role here is to moderate the session. The effort hitherto, as I say, has been bottom up. But now the project has advanced to the point where we now need to have conversation at the top of African politics for us, for the bottom up and the top down to ultimately start to come together. And in preparation for this session, a series of questions were sent to the ministers that we had hoped would be participating. And Dan and Professor Kobia, I hope you have seen those questions, because those will be the basis of that this conversation will be based on. And with your permission, therefore, Dan Dutoy and Professor Kobia, I'm going to start with the first question. And the first question that was sent is, what is your impression of the African Light Source Project? How do you see your country contributing to or benefiting from it? How do we best engage your government, universities, private sector, and civil society to progress the African Light Source Project in your country? Dan, can I start with you?
Thank you very much, Dr. Mukele. Indeed, and it's a great privilege for me to participate and represent my minister, His Excellency Dr. Nzimande, in this afternoon's discussion. And my first task indeed is to bring to all the apologies of Minister Nzimande. If he's not present here today, it's certainly not because of a lack of interest. Of course, as was indicated, as we speak, he is engaged in the intergovernmental affairs of UNESCO in Paris. And why it was important for the minister, if he was free to participate, and indeed why it asked me to represent him, is because an initiative such as the African Light Source Initiative, really is strategically aligned with a number of key policy priorities for the Department of Science and Innovation. First and foremost, and that is to harness scientific capabilities and to put those at the service of Africa's growth and development. But secondly, and this is where an initiative such as the African Light Source Initiative really come to the fore, and that is to bring African science together, to serve as an enabler, to serve as an integrator for deepening Pan-African, intra-African collaboration in science. And then thirdly, we, and Dr. Mukele spoke to this, really view large-scale mega science infrastructure projects as a key enabler to promote integration and cooperation in science and to be a powerful vehicle for developed capabilities, specifically human capacities. So the perspective from the South African government, from the Department of Science and Innovation, indeed is then one of an initiative which is strategically aligned with our policy priorities. It is an initiative we have watched and continue to monitor with great interest, and we continue to really appreciate the dedication and enthusiasm of many of the champions of this initiative present on this call, who over many years now have worked away. And I think that is the important message. And Dr. Mukele was too modest and did not spoke about his own critical leadership role, both with regard to the Southern African Large Telescope, as well as South Africa's eventual hosting of the Square Kilometre Array Telescope when he was the leader, the president of our National Research Foundation. So in the first instance, and what is required is for the scientific community, for the user community of these large-scale infrastructures, such as a light source, to consistently engage with government and to maintain a regular dialogue in order to make sure that our policy decisions and our funding decisions are informed so that they correspond to the needs and the priorities of our community, but also seek to strategically leverage the opportunities investment in these large-scale facilities bring around, because that is what happened now more than 20 years ago, when our astronomy community had engaged with Dr. Mukele and his leadership, such as the National Research Foundation. So in the first instance, an engagement such as we have today between policymakers, between the decision makers, and in the scientific community, the user community, is exactly what is required. Of course, it's very important that we have this discussion within an environment which is one where public funding is constrained. That is no secret, but that should not stop us from dreaming. That should not stop us from being ambitious, because I am convinced that there were many voices 20 years ago who would say that never would Africa, and of course South Africa is really hosting this for Africa, be able to host a facility such as the Square Kilometre Array. So I would also just like to encourage the group and this community to continue and to persist with its efforts. So I will conclude with those initial remarks. Thank you, Dr. Mukele.
Thank you very much. Thanks, Dan. Professor Kobia, over to you, sir.
Yeah, thank you, Professor Moketse.
Professor Kobia, we only see the top of your head. So if you can tilt your screen a bit so that we can see your full. Now you see, we can now also see your very elegant regalia now.
Okay. Thank you, Professor Katso. The email about this meeting has been addressed to the Director of Higher Education, the Director General of Higher Education, who went in France with the Minister of Higher Education, as we said, for the General Assembly of UNESCO. So from there, the girls told me to represent them at this meeting. They did not give me all the mail concerning that meeting to allow me to address all the question you ask me now. So I will do it later on by writing the response for each question, to what level my country, Côte d'Ivoire, will be involved in this joint conference and to this project.
Yeah.
And I would like, first of all, to tell you that in Ivory Coast year, our Department of Physics have an equipment dedicated to that's a kind of research. And we have some a team, an experienced team that consists of many professor and associate professor involved in such research for a long time. So we are very, very interesting to get involved in that project. And I will work with that team involved in the branch of crystallography, light source, to respond to all these questions and send back to you properly.
Okay.
That's what I'm going to say for the time being.
Okay, Professor Kobia. So
yeah.
I realize the slightly tricky position that you've been put in as a last-minute replacement, and so I certainly would not want to put you on the spot to respond to questions that you may not necessarily be ready and prepared to respond to. Can I propose, Dan, then that and Professor Kobia, that as I go through the questions, I will ask Dan the question, and if you have anything to add after that, Professor Kobia, I will leave it up to you. But if you don't have anything to add from the point of view of your readiness, we shall then proceed with Dan to the next question. I realize that you find yourself in a bit of a tricky situation. I think it's just unfortunate and fortuitous that this conference coincides with the General Conference of UNESCO, and especially with the Science Commission, which actually meets this week. Dan just reminded me that last week it was the opening plenaries, and in fact, Dan, you may want to talk to us a little bit about the Open Science Declaration that was adopted this morning. And Dan, talk to us about that. And after that, I'll then and just kind of share with this community what the significance of that is.
Yeah. No, thank you for that, Dr. Mukele. I think today is indeed an historic day for global science, and specifically also for African science, because I'm sure as many of the esteemed colleagues on the call would be aware, that UNESCO adopted a global recommendation for open science. And what does that recommendation mean? It means we have a set of rules, a set of policy recommendations, which will really seek to assist the global scientific community to unleash the potential of open science to make a contribution to the advancement of the scientific enterprise. And why this is significant is because whilst there's discussion on open science in many international multilateral forums, of course, UNESCO is the preeminent global forum which brings the entire global community together. And I think it's very important indeed that we reflect on that recommendation for the purpose of this discussion here, because the recommendations of open science very much speak to the enterprise of large-scale research facilities such as light sources or synchrotron. Because what is open science? Of course, it's first of all a scientific enterprise which is open in the first instance, inclusive, international cooperation. And that is what is also at the heart of this initiative, where we really seek to make sure that African scientists can optimally participate in global science by enjoying access to such important facilities such as a light source. But open science is also about open access. It's about access to open access to data. access to research results. And I think these are, again, also some of the values which we have seen is underpinning the African Light Source initiative. Of course, it is a research infrastructure, and the science conducted with it is data intensive. So the principles of open data, of open science, will be also embedded very much in the Africa Light Source initiative. But perhaps most importantly, open science also means science which is open to society, science which addresses, science which corresponds to the needs of society. And that's what we've also witnessed. I didn't participate in the earlier discussions, but I'm intimately convinced is that the motivation which brings all of us together here this afternoon is also to consider how crucial contributions the light source sciences enables us to make to address the needs of our societies, whether it's in health, whether it's in food security, or other critical disciplines. So I am confident that the decision of UNESCO to adopt this recommendation on open science, and then other initiatives which are responding to that, such as the African Open Science Platform, in which Dr. Mokele is also involved in, will really help us to create an enabling environment for an initiative such as the Africa Light Source to continue to progress. Thanks, Dr. Mokele.
Dan, thank you very much. And let me just add on to what Dan has just shared, that we have an initiative, the African Open Science Platform, which initiative was the pilot of that initiative was funded by Dan and the Department of Science and Innovation of South Africa. I happen to serve as the chair of the advisory council of that African Open Science Initiative. We've just recently identified the director to run the initiative on a 5-year grant, again, coming out of the Department of Science and Innovation of South Africa. This is truly a Pan-African initiative. I am co-chairing the advisory council with Professor Ismail Serageldin, who many of you on this call would know, former vice president of the World Bank, Egyptian national, who came back from the World Bank and re-established the library in Alexandria, Egypt. And we have on the advisory council a Kenyan, Sierra Leonean, a Scotsman, and the executive director of the International Science Council, and the executive director of CoData. And Dan is absolutely correct in the sense that what that initiative speaks to is exactly what the African Light Source project and initiative speaks to as well. So Simon and the team, as soon as the director of the platform starts on the 2nd of January, I will facilitate connection between that office and this initiative. Dan, you've kind of answered the second question already, but let me phrase it in full, because the latter part of the question is what I'll be more interested in. What is the biggest science facility or project in your country? I mean, clearly the SKA is the biggest project in South Africa right now. And you may want to talk a little bit about what that is. What is its annual budget? How is it managed? That is it government run? It's run via public-private partnership, or is it university-based? If you can just talk to us, Dan, about the SKA, its budget, its governance structure, and because it's a very complex governance arrangement that you've worked very, very hard to bring together, and you're absolutely the perfect person. Even if the minister was on this call, I'm sure the minister will call upon you to answer this question.
Well, thanks for that kind introduction, and Dr. Mokele. So indeed, the single biggest scientific infrastructure project in South Africa is, well, it's actually not the Square Kilometre Array. Technically, it's the Meerkat telescope. The Meerkat telescope, which is an official precursor for the Square Kilometre Array. And the Square Kilometre Array is really the ambition or the plans to build the world's largest radio telescope. South Africa, our National Research Foundation, again under the leadership of Dr. Mokele and our South African government, decided several years ago that in anticipation of the construction of the Square Kilometre Array, to learn valuable lessons, science lessons and engineering lessons, but also really to demonstrate Africa's capability, that we will build the Meerkat telescope. And I should say very proudly African that the Meerkat telescope is currently the most powerful instrument of its kind in that specific scientific category, and is already delivering world-class scientific results, which is making headline news in the international scientific community. Meerkat, at this very moment, is a national facility. So it's a national research infrastructure forming part of the facilities administered by our National Research Foundation. So indeed, it is fully funded by the South African government. In a few years from now, the Meerkat telescope will be incorporated as part of the Square Kilometre Array, the Global Radio Telescope. Now, earlier this year, a landmark decision was taken when the Council of the International SKA Observatory, which is the intergovernmental organization which is governing the construction and operation of the SKA telescope, made the decision to proceed with the construction of the SKA proper. So the SKA is now in practice. The construction work has started. Of course, I should say that this construction work builds on a decade, if not more, work of preparatory work, including an intricate design phase. And I notice with, I'm happy to see on the call, for example, we have John Wormersley, for many years a science leader at the Science Technology Facilities Council in the UK, and was actually the chair of the SKA board, because before the SKA became an intergovernmental organization, an international organization, we were a company incorporated in the UK, which was tasked with overseeing the design phase of the SKA project. And before that, it was really, there was different governance structures which brought together the scientific community, because this comes from the scientists. And I think that's perhaps a lesson for the African Light Source Initiative to progress through these phases, where you have a governance structure which is first and foremost resides in the scientific community, because that's what we had. We had a type of SKA science community. Then it progressed towards bringing the funders, the research funders together, the likes of the National Research Foundation, the STFC and others, who then created this company. But then when we came to the big decision to build a project whose construction budget will run into billions of euros, and then with enormous annual operational budgets of hundreds of millions of euros, because these are data intensive projects, these are energy intensive projects, we needed the firm intergovernmental commitment. And that's why the international intergovernmental organization was created. And there are also some technical reasons which we can discuss at another stage, such as, for example, it was very important for this large scale construction needed that we were able to design our own procurement policy, and we're not governed by any national legislation. So that's why the decision was taken to have the intergovernmental route. So very much the Square Kilometre Array will be similar to what many colleagues on the call will know, the governance structure of CERN. It's on the IGO model. It has elements of what some colleagues will know from the European Synchrotron Radiation Facility, but a large-scale project such as this kind really needs, ultimately, when it comes, not only the construction, but as all of those with experience of these large-scale facilities will know, it's the long-term commitment to the operational costs, which is the even bigger budget item, that you need to have a treaty, you need to have a convention which commits governments to those investments. So that is the trajectory of the governance debate. And I think what, from our perspective, we are here currently with very dynamic engagement from the scientific community, the user community, African scientists, the international partners. The next step, I would think, would be to start engaging with the research funders, so that we start step by step formalizing the engagement, because that's what we did with the SKA. We went from committees, we went from legally non-binding, non-for-profit associations, then set up the company, and today we have the intergovernmental organization. Because the day we progress towards that actual construction of such a facility, of course, then you need the strong commitment from governments involved. And again, something I said earlier, which I think is very important for projects of this kind, is to remain committed. It is a long-term initiative. The SKA governance, which led us from where we are today, it's a two-decade process. So it has to be, to conclude before I speak too much, always has to be, and I see John's hand is up and he may want to add to that, Dr. Mukele, I would recommend that, is that it always has to be a government which draw an initiative, a governance structure which is firmly rooted in the science, draws on the best scientific advice possible. That's why even now when we have an intergovernmental organization, on our governance structure, we have one government official and one eminent scientist from the country concerned, scientific advisory committees. But then to proceed to construction and operation, you need to have the formal commitment of the governments involved. So let me perhaps stop here, and perhaps John may also want to contribute.
John Womersley, comment, please.
Hi, thank you. And thanks also for the opportunity, and thanks to Dan for inviting me. So yeah, I think you have laid out a very wise approach there, Dan, because it's based on something that we know has worked in the past, and that's always a good place to start. We began SKA as a scientific initiative, but in fact, it encompassed many different scientific visions. And there were different scientists with different ideas, different levels of ambition. Nobody quite knew what it would cost, and nobody quite knew if it was achievable. So it was necessary, as Dan said, to organize some sort of design effort. And it was also necessary at the same time to set up what we called a funding agencies committee, which is the thing that could eventually evolve into a governance structure. But at the beginning, it was very much to have the sort of discussion that you are having today about judging the potential interest from each country, from the national research foundations, from the scientific ministries in the cases where that was relevant. And just to start a conversation, because we knew a conversation would take many years before it became firm financial commitments. But it would also take several years to design the project. And to do these things in parallel is essential. You need to understand the scientific needs from the bottom up, but also the funding and political realities from the top down. In particular, there are choices about the beam energy of a synchrotron. There are choices about instrumentation. There are choices about the how innovative you wish to be, which could have a big impact. It would be very tempting to build a facility that is the best in the world, but it might be very expensive. And these kind of judgments need to be thrashed out. It also makes it possible to become a family, to learn to work with each other, for the scientists to work with the funders, for the funders to work with the scientists, and to develop a common approach to taking the project forward. But it does require some funding, and the design phase needed to be supported, of course, a level much less than the construction. But to do a proper engineering design required that the funding agencies find a little bit of money, and the European Commission was also very helpful with this in the case of SKA with preparatory phase funding. So I think that's a very good approach, and it led in the case of SKA to a decision to go ahead with a full international treaty. But of course, that's a pretty slow process, and you can't get 7 countries into a room and tell them to sign a treaty until they have a very clear idea of what they are going to build, where it will be located, how much it will cost, and what scientific what science it will deliver, and the scientific and industrial relevance of that research. So I think for SKA, that process, I think for a light source, one could imagine moving faster technically, because many of these facilities already exist in the world. But I am not sure you can move faster politically. So so now is the time to start those discussions with the funders and to start the sort of structures that Dan has helpfully described. Thank you.
So John, thank you very much. And in fact, before I ask Professor Kobea if he has anything to add, let me also share this little bit of detail, John. that was it 2 years ago, when we assembled the Jodrell Bank. And Dan has just said that the governing council of the SKA has just given the green light for the telescope to be built. 2 years ago, when we assembled the Jodrell Bank, we are assembling 25 years since the SKA conversation started. So this decision taking now to actually build the telescope, incorporating the Meerkat telescope that is in South Africa and the technology that's in the Meerkat, gets taken 27 years after the initial conversations took place. I mean, that kind of speaks to the scale of a bottoms-up project that ultimately became top-down, and the two came together.
Yeah, but one shouldn't get too depressed by that, because I think it's more like 10 years since the first meeting with government representatives.
Yeah, absolutely.
So the timescale is not rapid, but it's within the career of those of us sitting around this table, I hope, at least within the remaining.
Thanks, all. Professor Kobia, any comment from you? You're muted, sir. You're still muted. I think I was told that I can unmute you, I think.
So His Excellency from Nigeria is just tuned in to the call, to the session.
I've just noticed that.
And also, Amadou Wage's hand was up for quite a while. I see it's down again, but you may like to ask him. He's the president of the African Physics Society.
Lawrence, can you unmute Professor Kobia? I think he's been trying to talk and...
I'm trying to, he needs to unmute himself there.
We can send him a message to unmute, but we can't actually unmute him.
Yeah.
I have been messaging him.
But let's cut to
let me take this opportunity to welcome His Excellency, the Minister from Nigeria, Dr. Ogbonnaya Onu. We were sensitized, Dr. Onu, that you'll join us a bit late, and we are happy to welcome you to this forum. We had already kick-started with Dr. Onu. Can you unmute yourself so that can you? I think you unmuted, sir.
Yes. Can you hear me?
Okay, go ahead. So thank you very much. We had already started, and perhaps of the questions that were sent to all the ministers that we had hoped to participate, but I can see from your body language that you may not have received the questions.
Yeah, it's okay. Just let us know where the questions.
But let's ignore the questions, Honorable Minister. Let me throw this second question at you. What is the biggest science facility in your country? What is its annual budget, and how is it managed? Is it government run? Is it run via public-private partnership, or is it university-based?
I must thank you so much, and I need to commend this effort, because Africa must pay attention and make sure that we deploy science, technology, and innovation to nation building, because all through history, no nation has ever become truly great without deploying science, technology, and innovation. So we have all the resources. We also have a very young population. We are very lucky indeed. We have water, we have very good weather. So there is nothing else except that will. And that's why I was very happy about this. And in Nigeria, for science, the public sector is making contributions. The private sector is also making contributions. And our universities in the area of research and innovation. As a matter of fact, in March of this year, the President Muhammadu Buhari, he actually allocated 0.5% of the gross domestic product of Nigeria to the science, technology, and innovation ecosystem. So we have a structure in Nigeria where the public sector, particularly the federal government, also some state government, own research institutes, research agencies, doing research in virtually all areas. But the Federal Ministry of Science, Technology and Innovation, we coordinate research and innovation in the country.
In fact, your response, Honorable Minister, taking us to another question that maybe I should swing that question through to Dan. In your case, you say the federal government has just committed 0.5% of GDP.
No, 0.5%, yes.
Yeah, 0.5% of GDP to science and innovation. Dan, let me swing this question to you and to Professor Kobia, because this is the one that Professor Kobia can respond to. Africa has been talking for many years about getting to the 1% of GDP dedicated to science. Firstly, where is your country done with regards to this 1% journey? And the second part of that question is, what do you believe is the hindrance of getting many African countries getting to that 1%? Dan, Professor Kobia, and followed by Professor Kobia.
Thank you, Professor Mokete.
Professor Kobia, let's start with Dan, and then we'll come to you.
Okay.
Okay.
Okay, you want me to answer, is that?
Okay, let's start with you then, Dr. Onu, and then we'll go to Dan, and we'll go to Professor Kobia.
No, I think it's very important. The African Union Executive Council in 2006 took that decision. And this 15 years ago. I think all African countries should really allocate a minimum of 1%, to GDP. Because if you look at the European Union, just imagine, just about 5 years ago, European Union requested all European countries to allocate 3%, to dedicate 3% of their GDP. And many European countries have done so, 3%. And so we know that the difference between the developed countries and the developing countries is merely paying attention to science, technology, and innovation. So I don't think I know that our economies, the private sector is not as developed as it should be. And there are so many challenges in terms of infrastructure and all that. But all African countries should try and make that sacrifice, because this is one way we can move many of our people out of poverty. China just announced that they have defeated extreme poverty, and Africa should be able to do so. And there is no other way except if we can ensure that we fund adequately, research and innovation.
Thank you, Honorable Minister. Dan?
Thank you, and it's an honor to continue on that intervention by the Honorable Minister. Now, first and foremost, where is South Africa? We're currently just below 0.8% of our GDP. We've had an objective for many years to achieve 1%, and in fact, some of our policy documents speak about an aspirational objective of 1.5%. The challenge has been that even though the government, even despite difficult economic circumstances, has been able to at least maintain our investment from public resources, there has been a decrease in investment from the private sector. And that is absolutely crucial. This is not a mission, an objective which can be achieved by governments alone, but governments have to work hand in hand with the private sector and create an enabling environment for private sector also to invest in R&D. And that has been a real challenge in the South African economy during difficult times, that we have seen that our investment from private sector did not match the investment which has come from government. And of course, there are reasons. I've already alluded to this, the difficult economic challenges, the fact that governments have many other policy priorities, which at first glance, and I emphasize the words first glance, could seem more pressing, could seem more urgent. So what is really important is to maintain this long-term strategic perspective, the long-term commitment, which is not aligned with cycles of administrations or electoral cycles, but really gets embedded in the long-term government planning framework. Because if you do that, that's when you see the results. We spoke earlier about a success story, which is South Africa's involvement in the Square Kilometre Array. And one of the reasons we were able to achieve those successes because our government has remained firmly committed to that objective. We've had several ministers of science and technology during the lifespan of this project who have always just continued building on the work. So the long-term commitment is absolutely essential. And then a vibrant policy dialogue within government on the role science and innovation should play in society. And it's very important that this is not a discussion like the ones we're having this afternoon amongst those who are converted, because we are preaching to ourselves, but it's that real engagement at the highest level of government with the ministers of finance, who are responsible for the budget decisions, and then really convincing the ministries with the other budgets. And that's what we see in South Africa really as our key role, is that it's our ministers, our sister ministries responsible for agriculture, for health, for energy, for trade and industry, which have far bigger budgets than we will ever have. And then we're not complaining about, but convincing them of the importance of investing in their portfolio, in research, in science and innovation, so that science and innovation is really mainstream as an instrument for growth and development. And if we do not do that, we will not achieve the lofty ideals we set for ourselves. So it also calls upon us in the scientific community to be outside of our comfort zone and to agitate and to convince others. Thank you.
Thank you, Dan. Professor Kobia?
Yeah.
Yes, so are you in Abuja?
So I was saying that it's an opportunity for us to get involved in this open initiative platform, African Light Source. But in our country, the research is entirely run by the government. Yeah. And with the university-based funds and some independent centers, depending on the Ministry of Higher Education. But all of these are funded by government. Yeah. What we are doing now is to interact with the private sector, to get funds from them and to try to make this sector more dynamic. So what we are doing is that we create what we call Africa Center of Excellence
Mm.
in mining, geology, waste processing, climate change, which is funded by private sector, international private sector, and which work well. We have these centers since 2 or 3 years, and the result is really, really, really interesting. But the GDP of what you are speaking, yes, we know our parliament members are aware of this goal of 1% of our GDP, but actually the real level is 0.35.
Yeah.
And when we present our budget at the parliament, they are aware of that. They said that, but the research is not funded enough. But we say, but we should the government to get to the goal of 1% of African Union said, but unfortunately we are actually on this level, which is 0.35. Meanwhile, the government makes some effort. Recently, we get some facility which is very interesting. We have the computer, which is the first powerful in sub-Saharan Africa after that of South Africa. And we are looking for a financial model so that we can interact, utilize this center with the private sector, and they are very, very, very interesting in it. But we are making the financial model to get together and utilize this computer. Meanwhile, university and some agricultural center and funded by the government or mixed funded private and government. But to boost our research sector, we create this center of excellence, which by the time, actually, presently is working well. So we are aware that the 0.35 is not enough, and we are looking to conduct our research in direction of the private sector. We want to know their need, and so that we can interact with the projects, the project that interests them, so that our research can be directed to the socio-economic world and get that funds. And we are revising our law to allow the university and center to interact with the private sector so that they can receive funds from this sector and make products that can be sold and get some fund from this sector, because presently all these departments are run by government. They are not allowed to take money from the private sector. So we are revising our law on research, on education, so that university, high school can interact with the private sector.
Thank you, Professor Kobia.
Yeah.
I need to move on to the next question now.
Yeah.
And this next question is designed to establish the extent to which the African Light Source Project is visible and alive in your countries. The 32nd ordinary session of the African Union Executive Council recently called upon member states to support the Pan-African Synchrotron Initiative, which is a term synonymous with the African Light Source. My first question, let me start with Honorable Minister Onu as well. To what extent are you aware of this call from the AU? And to what extent do you believe you may support placing the African Light Source Project in the STI strategy for Africa and Agenda 2063 as a flagship project?
No,
Okay, thank you, Professor Mokele.
No, no.
As I
Professor
said
Kobia,
at the beginning,
Professor Kobia, just hold on.
Yeah.
Just hold on. This is Dr. Onu first, and then I'll come to you later.
Okay, no, I make it very brief. I make it very brief. I think it's very important that two things that are very critical for us in Africa, because many of us, I think most Africans feel so unhappy each time we see Africans trying to run away from Africa to go to Europe. Many of them die crossing the Mediterranean Sea. We feel very, very unhappy because it shouldn't happen. You know, the resources we have in Africa, if we utilize them, the rest of the world will be coming here. And as a matter of fact, all parts of the world that have developed have done so using resources, you know, from Africa. And why can't we use the same resources, you know, to develop our own nations, so that we will no longer be the face of hunger, you know, because normally if you want to show the face of hunger in the world today, you need to bring an African face. Or the face of crisis and so on. All this shouldn't be there. Our standard of living should be far higher than what it is. I will understand what happened in the past, but we can't live in the past. We have a bright future. We should move there. So basically the point I'm making is that we cannot, one, African countries must work together. It is very important. When we share knowledge, when we come together, we will share knowledge. And by working together, we will, you know, learn from one another. And then we will not duplicate efforts. You know, we can, South Africa can learn something from Nigeria. Nigeria can learn something from Ethiopia. Ethiopia can learn from Ghana. You know, so that way, you know, our march to development will be faster. And it will be something that, you know, the same sort of thing that happens in Asia with the rise of Japan. You know, Japan developing immediately after the Second World War, and its impact, you know, being felt everywhere. You know, we now have so many Asian tigers and so on. So no technology. The second thing is that no technology should be, we should not, as a continent, you know, deny ourselves participating in any technology. We must get involved. We must. You know, and I tell people, I say, you know, when, you know, the oceans were being navigated and the ends of the world determined and so on, you know, Africans didn't do much. Though, we were, for example, Benin at that time, the Portuguese who came to Benin found out that Benin was so developed, you know, there are roads wider than the roads you could find in Portugal. But all those things didn't mean much, you know, because we did not participate. So we must ensure that no technology, I just want to make a general comment. Yeah. You know, no technology should be, you know, beyond us. We must involve ourselves, the emerging technologies, the old ones, our march, you know, this fourth wave, you know, technological wave, we must come in and make sure that we participate fully and, you know, understand what is going on and take over. It's possible and it should be done.
So, Dr. Onu, can I conclude from listening to you that Nigeria would be willing to champion the inclusion of an African light source into Agenda 2063 as a flagship project?
Yes, 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 yes. And okay. We're doing a lot. We're doing a lot in so many other areas.
Thank you very much. Professor Kobia, that's the question that I want an answer from you too. Are you aware of this decision by the AU executive, and is Cote d'Ivoire prepared to champion placing a light source project into Agenda 2063?
Okay, as I said at the beginning, our director of research, which is on mission now, was involved in that project. So he knows very well the background of this project, and as he's not there, he told me to join the meeting. But what I said is that anyway, it seems to me that as an opportunity to get involved in African platform, African light source platform, because we speak of African funding capacity, which is not enough to get individual facility. So getting such platform is an opportunity for the mutualization of our funds and capacity, and to go ahead. So for me, we are preparing to join this African light source, when my team will be back, we'll discuss and find the way to get involved intensively in the constitution of this African light source. But anyway, it's an opportunity for us, but because we are very, very, very aware that our general funds and the fund dedicated to research will not be sufficient to build something in for our whole country. And research has no frontier. So for the first time, if African can cooperate, be together, we cannot do otherwise that joining this platform, and we're preparing, as you said, for the year to come, to be actively involved in this African light source.
Thank you very much. Dan, same question to you, sir.
Thank you very much, Dr. Mukele. Of course, yeah, indeed, South Africa is very much aware of this decision. And I think the point I wanted to emphasize that this is already a decision which dates back to January 2018. So it's almost 4 years ago. Yeah. So what we would be more interested in from a South African perspective, rather than just broadly speaking about support, is to very practically, very concretely discuss with partner governments, but as well, of course, the scientific community. So how do we put this support in practice within a framework, within a plan which corresponds to the needs of the African scientific community? I think one should, it's important from an ambition, from a mobilization perspective, to use words such as flagships, but I think very importantly is actually to start, as we discussed earlier, step by step, build this initiative, and we would be open to discuss with all partners on how to take this forward. Thank you.
I'm looking at the clock. We have 30 minutes left in this session. I'm going to ask you the last question and then open it up for participants in this session to either ask you questions or make whatever comments they choose to make. When one looks at the two major projects that South Africa has spearheaded, the optical and radio telescope projects, these large pieces of equipment are dedicated to either optical astronomy and radio astronomy. We here in South Africa, we have a particle physics facility that's also managed by the National Research Foundation, which can do either both particle physics or the application of those particles in activities that go beyond just physics, radiobiology and cancer therapy and so forth. The beauty of a light source is its versatility in answering very specific questions in health, in agriculture, in sustainability, in the environment, and so forth. And the COVID pandemic that has shaken the whole of humanity at its roots has reflected the importance of structural biology as an area that a light source facility can make fundamental contributions to. And so my question to the three of you is, how does the evolving response to current and future pandemics, how should it shape Africa's policy in respect of the African light source? If you understand what the light source facility can do, but you know, how do you envisage it shaping Africa's policy to respond to having its own light source facility to enable Africa to respond to these pandemics and other crises? Dan, let me start with you before I go to Dr. Onu.
Thank you very much, Dr. Mukele. I think one of the lessons we have learned, or what we have seen in the South African scientific response to COVID-19, has been just how it has emphasized absolutely the critical importance of long-term investment in research infrastructure. Because during the pandemic, as we sought to mobilize the South African scientific community to respond to COVID-19, we were able to draw on a number of strategic capabilities developed through research infrastructure, such as, for example, our Center for High Performance Computing, which was critical for all the big data modeling and analysis required to inform government's policies responding to COVID-19. Similarly, the extensive networks to conduct social surveys, which has been established, was very important in order to develop an understanding of the South African public's response and understanding of COVID-19. Or perhaps just to use the example of the square kilometer array of what we have spoken about, when our government at very short notice had to develop our own ventilator manufacturing program, we turned to where we could find the best engineers in the country. And the best engineers were those who were actually responsible for the Meerkat and the square kilometer array radio telescope. And they were responsible for the national ventilator program. So these are all examples about how these investments, either directly, and you mentioned the example of structural biology, or indirectly through building generic advanced scientific and engineering skills, are important to prepare Africa for the future pandemic, or indeed for other global challenges, such as those related to the climate crisis, such as those related to food security, to which us as a continent has to respond to. So we have absolutely no doubt that investment in structural biology are one of the key disciplines. It's prioritized as part of the South Africa's new decadal plan, and in order to optimally draw on the capabilities of that discipline, we need access either to global research infrastructures, or preferably access to Africa's own infrastructures, such as a light source. So for me, whether it's a pandemic or other global challenges, these are the type of investments we as a continent certainly need to make. Thank you.
Thank you very much. Honorable Minister?
Yes, I really thank you. I had wanted to mention to you that I thought I would spend just 30 minutes, with this because of other engagements. But I'm very happy, exceptionally happy for what I am seeing, this level of cooperation between African nations. I think this is something that we must encourage. It's important that it's done at the political level and also within the scientific community, because it will really accelerate the pace of development. Yes, just about 100 years ago, slightly 100 plus, we had a pandemic, and now it repeated itself slightly after about 100 years. So we must prepare for the next one. It must come. It may not be in our own lifetime, but we have to prepare for future generations. And the way Africa was found at this period is something that is not very satisfying to us because, you know, as of today, we still have to rely on others to give us vaccine. And we're talking about vaccine nationalism and so on. That shouldn't be where we should be. We should be able to produce our own vaccines about the same time that others did. Yes, you know, the head start may be different and all that, but just like it's been mentioned with South Africa, yes, you know, you have to have long-term investment, you know, in order to achieve this. And it can't, you know, your research must be both basic and applied, you know, because we have to be looking, like I said earlier, we're just trying to talk generally. You know, we cannot be left behind in any area of, you know, science and the technology. We're seeing that biology is going to play a very vital role, more role than it played in the past in this century, and perhaps even beyond. We know the role that physics played, you know, with the atomic bomb and all those and so on. So biology is going to play a very, very important role. So this light source is important. And other emerging technologies, we must embrace them. We must build up the capacity, you know, within Africa. You know, Africa should be ready to start solving African problems by themselves. You know, we have some of the brightest people who are African, who are in, you know, research centers, universities in so many countries of the world. You know, we're also in Nigeria, we're trying to bring them home, you know, so that they can participate in making sure that we're ready because there are so many problems to be solved. Take, for example, Nigeria with over 200 million people. We have to feed that large number. We have to clothe them. We have to house them. We have to make sure that they go to school. We have to make sure that, you know, when they're sick, you know, we look after them. So, you know, these are challenges that must be met, and we must secure ourselves, you know. So the key thing that Africa should know is that, you know, the time has come for Africans to try to solve their problems themselves. You know, yes, we can always get support from our friends and partners, you know, outside Africa, but we must have the capacity, you know, to be in a position to solve many of the problems that are there. And the pandemic, yes, this pandemic, science is still studying it, and we can see the spike that we're getting in Europe, the shutdowns, even in China and the many European countries. So it is still, you know, science doesn't yet have a firm grip, though there is improvement, you know, but the firm grip is not there. But definitely, if we had a pandemic about 100 years ago, it is only wise for us to think and know that there will definitely be another one in future. Thank you so much.
Minister, so did I understand you correctly? Are you going to be leaving us now?
Yes, I have to, because I have other engagements. I would have loved to stay longer, you know.
Before you go, Minister, then let me perhaps make the statement to you that those of us who are championing this continent having its own light source are motivated by exactly what you've just said now. We're motivated by the fact that, yes, this continent has to continue to be part of the global commons, but it must also, not just for the purpose of solving its own problems internally of the continent, but it must also be a useful contributor to the global commons.
Oh, yes.
This continent has for far too long been the consumer of knowledge generated by others to solve its own problems, and not even having enough human capacity to consume that which is generated by others. And an African Light Source Facility, Minister, is a very versatile project. As you and Dan have indicated, that can be relevant to the structural biology that's required for vaccine development, that can be important for food security projects, that can be important for sustainability projects. And there's a whole host of projects that are at front and center of African challenges, that a facility like this with its versatility will be a frontline facility to help the continent address. And my last statement to you, Minister, before you go, the European Union and the European community that decided on 3% of GDP going to science, or Japan post-World War II, those nations, Minister, made choices. They made choices against many competing priorities and chose to prioritize investment in research, development, and innovation. This continent continues to resist making the choice to prioritize research, development, science and technology and innovation. And that's the culture on this continent that needs to change. Thank you, Minister.
Yeah,
we are delighted that you could join us, and hope you'll hear from us in the future.
Thank you. Thank you.
Thank you, Minister.
Oh, I'm bye.
Professor Kobia, your turn, sir.
Yeah, thank you, Professor Mukele. But as you said, we need some this light to source community to make pressure to our local governments and to make them aware that research, as you said, is a choice. A country like Israel, a country like South Korea, a country like Japan make the choice to dedicate their GDP around, let's say, but a consistent part of the GDP to research. And the result is there. We can see South Korea, we can see Japan, we can see Israel. We could do as this country. That's why an African light source, it's an African community to build our capacity, and it can be an instrument to a facility to make pressure to our government globally to make them aware that we have to push ahead our GDP in the research. Because if we are individual department of research of ministry, it's not enough to make this pressure. That's why I take this as an opportunity to be together and have a weight to make pressure to our local government, and our involvement in such a community like African Light Source is a real opportunity to make, because we have a capacity to make research like this country. The challenge of COVID-19, individually taken, African can do nothing, but together we can face the challenge. Many predictors said that Africans should be devastated, but it's not there. And we must understand why the people things work like that. And if we have such a community, it will be nice for us, an opportunity to make pressure to our local government and go ahead with our GDP in research. That's what I would like to say.
Thank you, Professor Kobia. A country that I would add to that list of countries that you've spoken about is Cuba, which is otherwise a very poor country, but that has always understood and appreciated that at the front line of its development agenda is science and technology and innovation, is research and development. and and prioritizing under incredibly difficult political and economic circumstances at times. And knowing that the more difficult those problems are, the more reliant they would be on research, development, and innovation. Thank you very much to Dan, Minister Onu, and Professor Kobia. We have 15 minutes left. Let me open it up to, you've been quietly listening to these three gentlemen. Any comments or questions from any of the participants? Please raise your hand. Hopefully we shall be able to see who's raised their hands and call on you.
Okay. I just want to put your attention to Prof. Amadou Wage. He's had his hand up.
Over to you, Professor Wage.
Can I speak?
Yes, you can speak, sir.
Okay, thank you very much. First of all, I would like to apologize. You know, this morning I was not able to join the opening ceremony where I was supposed to deliver a speech for 5 minutes. I am really sorry for that, because as the president of African Physical Society, which is one of the organizers of this joint conference between African Light Source, between Pan-African Crystallography Association, and also between our African Physical Society, it was really very sad for me not to be able to be connected by the Zoom. It was, I think, some technical problem, not internet problem, but just I don't know what happened. But after I was obliged to make a new registration to Zoom, so that I was able to join now. So thank you very much. And I just took the train in March, and I was very, very impressed by what I have just listened. The Minister of Science from Nigeria, the representative from Ivory Coast, and the others, and yourself, and also the representative from South Africa. And I am happy to hear that the Minister of Nigeria says they want to join the project of African Light Source. And I am very happy also to know that people now are aware that synchrotron light source is very important for the development of Africa. For the too many applications in physical science, in medical science, in industry, in et cetera, in material science, even in fundamental research. So this sounds very well, and I hope that this will continue, because I remember I was in 2003 in Johannesburg for the African Ministerial Forum on Science and Technology. And at this time, the African Laser Center was supported by this ministerial conference as a flagship program of NIPAD. And there was a promise that every country, that all the African countries together can give about $20 million per year for this project, so that all the nodal in Africa will have hard infrastructure in laser technology and laser application. But working today, it is only South Africa give a contribution to this project. But we are still hoping that the other country also can join. So and I really hope that concerning the African light source, because we are now in 2021, many things have changed. And now all the nations are now, all the politicians, all the governments are really aware of the importance of science and technology. The COVID-19 pandemic shows that without science, without technology, the humanity will disappear. So I think that the African government also, the African politicians are all aware of that, and that they will really contribute for the development of this project of African light source, and that before, I am not going to say 2063, this is very, very far. I hope that before 2030, maybe we'll start to build our African a synchrotron light source. Okay, this is what I want to say, and thank you very much for giving me the floor.
Thanks, Amadou. Any other comments or question to the two panelists?
I wonder if I can make a brief comment, please. Otto?
Okay, Simon, over to you, sir.
I just want to make an observation that we did have 3 ministers here, and we were not successful to have more. At times there were more. And my observation also is that the Ghana vice president was also due to reiterate the stance of the president of Ghana, that Ghana also committed itself to champion the African light source. So would like to thereby include it almost in the minutes or recording here. There have been continual conversations with the Ghanaian Science Ministry, and this dated since the conference in 2019, which was held in Ghana, and also Ghana did come to the conference in 2020. I think, as Khotso mentioned, it's very difficult for ministers to have solid diaries. I fully understand that. But I do want to say we've had a similar sentiment from the Ghanaians, and this morning also from Benin. And Benin have actually made Semi-City as a regional science center. And also there are many African countries where we experience things like this. In Rwanda, there's also a regional African science center. And now we also find from Botswana and so on, I'm sure many more. And I think it could be so helpful if the ministers present here and people with access to this top-down conversation, as Khotso has mentioned, I think this has been very important for the top-down conversation. And I really believe there's almost like a wind blowing through Africa to echo the words that we heard from the Nigerian minister. I'm sure we'll hear similar from the South African minister if we could, but definitely where Africa wants to use science, technology, innovation to drive a socio-economic transformation and generate wealth and be able to really realize this vision. All the points I heard, I absolutely like that we are a young continent. We will soon be the most populous continent, and these are all of our strengths. And last thing, I don't want to take up too much time, but I also heard the words from the Benin minister that also the African Union Commissioner said is leave no country in Africa behind. I think it's very, very important that the light source is not winner takes all, but we have an improvement of human capacity building everywhere, and also that we have many light sources, smaller scale facilities, such as we've heard in this discussion in very, very many countries in Africa, so that we really have solidly leave no country behind as we progress towards the light source.
Thank you, Simon. I'm going to take 2 more comments and then we'll close this one. Kenneth Evans Lutterot, if I'm saying it correctly.
Yeah, you are. It's good to see you again. My question is a little tiny bit of background. So it's very clear as day that improving the investments in science and technology and the light source constitutes such an investment that it improves society. And as was mentioned in this session, South Korea, China, and I think you also mentioned Cuba. All this leads to a better society. So the question is, how can we communicate this message more clearly to the political class? Yes, it's true. They seem to be waking up to this issue, but I'm not sure we're making the best arguments. So that's my question to everyone.
Okay, let's take that question and as we digesting it and the last comments, I'm very conscious of pronunciations. Alwase Ogweno.
Okay, good. It's evening here. Good evening, everyone. I'm joining from Mombasa, Kenya. I heard Dr. Mukele talking about how developed countries have improved in terms of investing their GDP into the research, a substantial amount. However, we in Africa and our African countries are still pulling their legs in terms of embracing such kind of intervention, in terms of building up on research and also ensuring that development from that aspect continues. It's very perturbing that not even a great pandemic as COVID-19 can move them that fast. So we hope that things will change, yes, because of what we've observed and what we've experienced. However, it might not move that fast. It was my comment or maybe suggestion that the panelists who are composed of ministers from different countries here may intervene in a small way for a start by lobbying the governments that are already signing into the African light source, just in case they can accommodate other researchers, because from other countries we have people who are interested in research, but they cannot mobilize funds at the level of a nation. That is, if their governments have not embraced joining or supporting such kind of joint ventures, so that such people can be given access in terms of research and being able to access such facilities to just enhance. Maybe in the long run, their governments will also embrace and then put in some funds in order to Africa to come together in terms of research. That's my small suggestion. Thank you.
Thank you very much. Malik, I see your hand, but I'm not going to recognize you, unfortunately, because I'm mindful of time. I just want to give Dan and Professor Obia an opportunity to make last-minute closing comments. But in your closing comments, address the question that Kenneth asks. How can we make a convincing case for African politics to recognize the light source as a crucial instrument for African development? Professor Kobia, let's start with you.
We also in all the meeting with our parliament member, we make them aware that our GDP and we have some capacity to make research and a good research, but we need a fund. And they said they are aware of that, but they are not, they can push the government to go further. That's why our commitment in the African light source will be an opportunity, because if we are together, it will create a lobby to understand our government that we are interested to have the same capacity of a European, as Asian, as American. But we need your involvement with funding, with everything, to realize the same thing as this developing country. So for me, we have to build this community, which will be an instrument of lobbying and to make our community, our local community understood by our government to push this GDP a bit further to build a real capacity and to make our development. That's what I would like to say.
Thank you. Dan, closing comments from you, sir.
Thank you very much. Just in terms of the communication, again, what we have said before, this should be an ongoing effort. It should always be focused on the societal challenges, the economic challenges we seek to respond to through science and innovation. But I think it's also important for this community to really highlight to the policy and decision makers the enormous potential this initiative had for really practically reinforcing Pan-African cooperation, Pan-African integration. We know that we speak a lot about Pan-African cooperation, but if we look at the patterns of scientific collaboration, most of it is still North-South on our own continent. So we need programs, we need platforms to really meaningful develop Pan-African programs, which also serves as catalyst for human capital development. So it's really this sort of holistic vision of a vehicle which can advance the Africa science agenda. And I think then just most importantly is to remain committed and not to be discouraged. Thank you very much.
Thank you very much. Let me take this opportunity to thank.
So before, can I just tell you, we have the AU just come in here. Is Her Excellency the Commissioner of ESTI just joined?
So
Sorry.
Give me guidance now. What should I do now, Simon?
I think maybe she could have a few minutes. I'm not sure. Or you are the chair, so you should do it. But I just want you to be aware that Her Excellency, the AU Commissioner, has just come in.
Okay. Your Excellency, Dr. Sarah Agbo.
Yes, sir.
I can see you.
I'm so sorry. I've been trying to log in, but it's like I'm not an Android girl.
Okay. You've just missed a very lively session. We heard from the Nigerian Minister, Dr. Ogbonnaya Onu, who unfortunately had to leave us a few minutes ago. The Minister from South Africa, Dr. Blade Nzimande, is currently at UNESCO, and so is the Minister from Cote d'Ivoire, with the Science Commission at UNESCO starting this morning. And so, but they had representatives who were on this call, and we're just about to close because the session was scheduled to close at 7. But I'm happy to give you 5-10 minutes to hear high-level thoughts from you on how the AU Commission sees this African light source project.
Thank you very much, sir. I stand on existing protocols. Good afternoon, distinguished ladies and gentlemen and excellencies. It is a great pleasure for me to join you in this third African Light Source Conference to discuss and collectively explore ways on how to realize such a far-reaching and ambitious initiative, which is important for the development of science, technology, and innovation in Africa, which is the mandate of my department, the Department of Education, Science, Technology, and Innovation. Let me also convey warm greetings from His Excellency, Dr. Musa Faki Mahamat, the chairperson of the African Union Commission, who is equally seized with the socio-economic development for the Africa we want through our long-term Agenda 2063, which cannot be fully realized without vibrant, strong, and sustainable science, technology, and innovation capacities and platforms that empower the scientific communities to unlock potential solutions that address our development challenges in health and diseases, beneficiation of our natural resources, such as minerals, agricultural products, climate change, and the environment, among others. Excellencies, ladies and gentlemen, the African Union Commission deeply believes that investment in STI capacities is critical for our research and innovation ecosystems. Our continental STI strategy that we commonly call the Science, Technology, Innovation Strategy for Africa 2024 identifies 4 pillars for building science capacities at the national, regional, and continental levels. These pillars identify with AfLS conference thematics. I will emphasize on the first and second pillar. Pillar 1 focuses on building, improving scientific infrastructures. We need centers of excellence for research and innovation, laboratories and world-class STI infrastructure and facilities for conducting research, teaching and learning, engineering, testing, prototyping, and clinical trials among these for technology, diffusion, transfer, and uptake. We should improve connectivity, ICTs, and digital infrastructure that enable collaborative research, access to data, and co-creation of knowledge. Pillar 2 focuses on improving technical competencies through education underpinned by the Continental Education Strategy for Africa 2016-2025. Africa has to spare no effort on its human capital development with special focus on women and youth. Africa's higher education must be attractive and research intensive, thus contributing to the acceleration of our continent's transition into a knowledge-based and innovation-led economies. Distinguished ladies and gentlemen, a synchrotron is one of such enabling infrastructures, a very powerful scientific tool with potential applications across many development sectors, health, industry, material sciences. The initiative on African synchrotron has already reached the ePCCr during its session of October 2017 held in Egypt, Cairo. In their decision, the ministers called the member states to support the Pan-African Synchrotron Initiative. The ministers are therefore seized on the matter, and I'm very happy that at least 3 ministers joined this conference and spoke at this high-level discussion. And I also use this opportunity to call upon all the relevant stakeholders, including the African Academy of Science, to work together to realize this important initiative for the continent. The African Union Commission will continue playing its role to ensure political support for the creation of such critical continental research infrastructures and inclusive participation of all our continental institutions and our development partners. As you have rightly said, sir, the discussion so far has been very interesting. And I believe that the outcome of this conference is very important, and we look forward to sharing it with all member states during the next STC-ESC, that is the Science Technical, the Specialized Technical Committee on Education, Science, Technology, Innovation that will take place mid-December this year. So we want to thank you and want to congratulate you for setting the tone. And we wish all stakeholders the very best. Since we are not going to meet before the end of this year, I take this opportunity to say Merry Christmas and the super abundant prosperous 2022, sir. Thank you.
Excellency, thank you very much for those encouraging words. Very encouraging to learn that African ministers and the Commission are already seized by this project to locate a synchrotron light source on the African continent. I can promise you that we shall work together with the African Academy of Sciences to make sure that Africa speaks in one voice when it comes to this particular initiative. Rest assured that this global community, because the community in this meeting, Her Excellency, is African, but also we have partners of Africa in North America, in Europe, who are also wanting to be part of this project as we locate a versatile facility that you've described, that's a synchrotron light source. And this community will be very eager to work with you and your team in the Commission to realize this very important project. Thank you, Her Excellency. And we're glad that the session dragged on a little longer than it was actually scheduled to, and that we're actually able to accommodate you to come and join. I know that Simon and others were going to be crucifying me for having let the session run so long. If only they knew that I was waiting for you to join before I could close the session.
Thank you, sir. Thank you very much. Thank you. Congratulations.
And thanks, everybody. I'm delighted to see that, people stayed on this call, even though it went way beyond time. And it just signifies the degree and level of interest that we all have in realizing this very important project. With those words, I say good night to you and those who celebrate Christmas. As the commissioner said, have a good Christmas. Those who do not, have a good rest of the year. Thank you very much.
Thank you, sir. Thank you very much.
Thank you, Professor.
Okay, thank you, everyone. So the conference will resume tomorrow morning.
Bye-bye.
Bye-bye.
Bye.
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 Bye-bye.
Thank you.
Thank you. Bye-bye.
Thank you, everyone. Bye-bye.
Bye-bye.
Thank you. Bye-bye.
Thank you. Bye-bye.
Bye.
All right. Thank you. Bye-bye.
So just
Is the commissioner still listening? Commissioner, are you still there? I think you're still muted.
Yes, I'm still there.
Oh, hi. I guess, you know what, I didn't want to add, I guess I wanted to say farewell, because I know this is your last probably 2 weeks before you leave.
Yes, sir.
But you're not leaving us. You'll still be involved with us, and we'll still be able to call upon you.
If you want to make me your consultant, take me. I'm available.
Make sure we have your email.
Yes, I will send you my personal email. Maybe I can put that in the chat. Thank you very much. I'm so sorry.
Congratulations for a nice tour of duty, for being the commissioner, and we just hope to see you soon.
Thank you very much, my brother. I appreciate it. Thank you very much.
All right, thank you.
Thank you.
Vâng, khúc bê cả.
Richard, are you still there? I see Richard. Kenneth, are you still there?
Hello.
Hey, Kenneth, how you doing?
How you doing?
Doing all right.
What's happening? Hey, you guys are doing a great job, by the way.
You're at the office. It looks like.
Yeah, I'm at the beamline.
Yeah.
You guys have done a great job, by the way. I should let you know. You guys have done a fantastic job. I know it's always kind of panicky and a mess, but you guys have done a good job.
Yeah, anarchy is a good word. I was like, this is not what you said you would do an hour ago. But I guess, they get calls to do stuff that You're lucky you can get them to sit down for, you know, any more than a half an hour, I guess.
It's amazing. Yeah.
Yeah, I guess it, I don't know. And then I guess people, I've noticed that the Zoom app works better for some platforms than others. So people who are using the Zoom app on Macs seem to be having a lot of problems. And I think I know why. Because I think because we're using breakout rooms. So apparently that requires you to use the latest version, the latest update of the app. And then we have people trying to sign in by their phones instead of their desktop. And I think that is problematic for some people. Anyway, lots of good science today.
Yep.
And we have some good talks tomorrow too.
Okay. I'll tune in.
All right. So talk to you guys later.
Yeah, it starts what, 8 a.m. South African time. So what time is that for us? Because I know it was beyond when I woke up.
I think the first talk tomorrow is maybe 9 South Africa time. I think it's closer to 9.
Okay. Take a look. See if I can wake up.
All right.
Okay. Take care.
We'll talk to you later.
Yep. Talk to you. Thanks again.
Is that Nellen Tamba still there from the AUC? I see you're still here. I want to thank you for making sure the commissioner got here. And let's see. I think we're all good. So I'm going to end the meeting.